Welcome all participants on the day 4 of this GIAN course on environment contaminants. So today in the first session, we have Dr. Shiv Kumar Malik from Department of Chemistry, Punjab University, Patiala. He has been basically the instrumental in starting this course or bringing this GIAN course to Punjab University, Patiala. And he's also local coordinator of the GIAN in this university. Now, Dr. Malik is an eminent scientist in the field of environmental chemistry, particularly environment analysis. He has many publications and pre-concentration methods in increasing sensitivity of analysis using various techniques for determinations of different kinds of contaminants in environment. And he is a regular visiting professor from our university to Spain, to Germany, and Australia. And he is a permanent fellow of some universities also. So he is a renowned scientist in environment analysis. So we are fortunate that he is with us. So now I request Dr. Malik to start with his presentation. So. Dr. Malik, over to you, please.
I am audible?
Yes, sir.
Okay, thank you, Dr. Dear participant, dear Professor Gulshanji, and my dear participants. Thank you for the Dr. Professor Gulshanji. Thank you for the introduction and topic of today I would like to discuss is pre-concentration methods for quantitative sensitivity enhancement. Now, this course was framed in such a way that first you get introduction to basic environmental chemistry or environmental science, that what are the pollutants, what are the different factors which are affecting the pollutants, how they stable, are they stable in the environment, how they degrade in the environment. I think this was necessary and you are already familiar with these aspects. Second aspect was yesterday, like Professor Gulshan told you some about instrumentation, mass spectrometry, and Professor Yolanda in detail gave you a discussion about mass spectrometry. For any analysis or sample characterization, you know, two things are very important. First, we should find the sample. What is the sample? You have learned about it. It should represent the whole community. It should represent it should be a representative of the community, that it of the total environment. And though it is very difficult that a small sample can be a representative of the whole unit or whole environment, but that's why we have to carry out a number of analysis and a number of collect a large number of samples. Now, samples, they are very widely distributed in nature, like you have food, cosmetics, biological tissues, and various environmental samples, and like titanium dioxide, silver, iron oxide, silica, gold, and many nanoparticles. They are present in the environment also. And for the analysis, you can say the particle size distribution, concentration, composition, and shape. All these are affecting the distribution of sample in the environment. So if you want to carry out direct analysis of the sample, that is quite difficult. For this, we have to carry out dilution and phase separation, extraction and matrix decomposition, and pre-concentration of the sample has to be done. So sample preparation is a very important aspect for an analytical chemist. Without proper sample preparation, without proper sample, you cannot carry out analysis. Analysis is totally dependent on the method of your sample preparation. Now, there are many sample preparation techniques for environmental samples or environmental analysis. So first is like adsorption based, or you can say these are the solid phase extraction, solid phase micro extraction. And there is another, I will come to this later, liquid extraction, matrix, solid phase extraction, and functional materials, computational modeling. This can also be done for the extraction, and hollow fiber liquid phase extraction, solid phase micro extraction. And out of these solid phase extraction is very important. I will not be discussing solid phase extraction. I will give you a very brief introduction, and Professor Yolanda and Professor Gulshan has also discussed with you solid phase extraction earlier, little bit. But tomorrow, Professor Olak, Jatinder Singh Olak, he will be dealing with you with solid phase extraction, different aspects. He will show you how to extract, do the extraction, and how to proceed further with you will have a virtual lab tour in his, he is arranging for this presentation or lecture. So I will give you a brief introduction to the different pre-concentration methods and how we can apply these methods for the analysis. Another thing is what are their advantages and what are their disadvantages, in which sample we should apply which technique, and we will discuss these things. QECHERS, this is a quick, easy, cheap, and very simple extraction method. This should be done very quickly, very easily, very cheap, very easy, responsive, and sample preparation method. These are the requirements for modern sample preparation. Now I will come to what is pre-concentration. pre means before, and that is concentration. Means increasing the concentration of analyte in the sample. Why it is necessary, and IUPAC has defined pre-concentration process. Let me say, pre-concentration means to increase the concentration of a sample prior to analysis or detection. For example, you can do pre-concentration for a non-volatile sample with evaporation of its solvent. Now, like if the sample is non-volatile, we can allow the solvent to evaporate, and it will increase its concentration. So pre-concentration means to increase the concentration of an analyte in the sample. And this is an operation or a process by which micro components are transferred from the sample of larger mass into sample of smaller mass, so that the concentration of the micro components is increased. Means it is the increase in the concentration of a component from a large volume into smaller volume. And various examples like decrease in solvent volume, distillation and transfer of micro components from an aqueous solution to a smaller volume of organic solvent by extraction. This is the definition given by IUPAC, that we transform a micro component. Micro component means a smaller samples, means which are in very low amount, into a micro concentration range. So like for an example, like this is a water, we evaporate it. These are the molecules, analytes. And when we evaporate the solvent, it decreases. It comes to a lower it comes to lower volume, and overall the concentration is increased. So this is pre-concentration. That is, pre-concentration involves evaporation of the solvent, or we are extracting, we are removing the analyte from a large volume area into a small volume area. Now, in chemical analysis, for chemical analysis, 3 steps are very important. First is sampling. Sampling, you already know that we have to convert, we have to extract, we have to collect the samples, and then we have to prepare the sample with, and we can prepare the sample with heat, acid, dilution, depending on the suitable method. And after that, we carry out the analysis of the prepared sample depending on the characteristic property of the analyte. Means whatever, depending on the suitability of the analyte, we carry out the analysis. So 3 steps are very important for the analysis. First is we should collect a sample, we should prepare the sample, means we should extract whatever we want to do, and third, carry out the analysis with a suitable instrument, means whatever may be available, or depending on the characteristic what we want to know about the sample, we carry out the analysis. So these three steps, they are necessary for the characterization of a sample. It may be any sample, means it is not necessary that it may be a soil, it may be a water, it may be contaminants, it may be a sediments, whatever it may be, but it is very important. We must prepare the sample, and from the sample means we should extract, we have removed all the interfering impurities from the analysis, from the analyte. Means we try to remove most of the interfering impurities from the analyte, and then we carry out the analysis with a suitable instrument. Now question arises why we cannot put the extracted or the prepared, the sample we have taken from the environment directly into our instrument. If this is possible, we will save a lot of money. But normally we find that this is very difficult. The analysis is not possible. Means without the preparation of the sample, we cannot, it is almost impossible that we can inject the sample directly into our instrument. That is, we take the sample and carry out, we collect the sample and carry out the analysis. This is almost impossible. So therefore, we need to prepare the sample in the best possible way, and our results will not be representable. Means we cannot conclude anything if our sample preparation is not sufficient or is not very good. Sample must be, the prepared sample must be very clean and suitable for the analysis. Clean, I mean, it should contain the analyte. It should not have most of the impurities which are present in the sample. It should be very, very analyzable. It should be an analyzable sample. So another thing is very important is, as we were discussing, why we should prepare the sample, that there are some particulates and in the sample, and they may limit the instrument system, and plugging may take place because these are some precipitated things or some undissolved materials, and they cannot be transferred to the instrument, and they make a complication in the system. And if instrument is sample is not analyzable, then we will have less complicated analytical results. We may have a less sensitivity, and there may be ion suppression, and it may reduce instrument system setup time, or we may take a very longer time for the analysis. So this is very important that we should prepare the sample in a very better way, like in mass spectrometry or in HPLC. If you take a sample, water sample, and injecting it into the MS or into uh, with any detector system, that you will get a large number of peaks, and you don't need these unwanted peaks. Means you need only these peaks which are corresponding to your samples, which are corresponding to your analytes. So it is necessary that you can say this chromatogram or this spectra by which we cannot conclude anything. We should get a clear spectra. We need only to analyze these peaks, we should characterize only these peaks corresponding to the sample, corresponding to the analytes. And this dirt, or you can say this unknown peaks, which are not corresponding, corresponding to our sample, which are not corresponding to our analytes, we cannot do anything with such things. So there is need for sample preparation in order to have a clear representation, or in order to carry out the clear analysis. Another thing, as I told you, I will show you that if we carry out the analysis, that during this is a HPLC chromatogram, and you must have, if someone has done HPLC, you will see that there are small peaks. This is a blank sample, and this peak and this peak and this peak, which are not corresponding to our analyte, are appearing in the sample. They are useless for us. They are spoiling our instrument. We don't need these peaks. We don't know what are these samples, unknown samples, and we are not. We don't need these peaks, and for this purpose, what we carry out, that we carry out SPE, solid phase extraction, cleanup. And after the solid phase extraction cleanup, all the baseline interferences, most of the interferences, they are removed. Like in this peak, there was this small, you see this interference is there, but in this here, after the removal of the SPE, after the removal of the interferences, you get a clean peak corresponding to your analytes. So this is very important, that we should get a clear-cut baseline for the sample. And this, you can say that cleanup of the sample in order to remove the interferences is very important. So another thing is very important, that if we are carrying out the analysis, the sample which is present in a very low concentration, without, like if we are carrying out analysis without pre-concentration, like this is a small peak, it is appearing, and our instrument is not in a position to detect this peak without pre-concentration. And if we carry out pre-concentration, we can have such a large signal, and we can carry out our analysis in a very easily way. So SPE or any other method by which we can convert this analyte, we can concentrate this analyte, and we can get this type of the peak will be very important. It means we are increasing the concentration, or we are increasing the response of the sample for carrying out our analysis. And you will say that if we can, like this peak is, it is almost in the noise level, and we cannot detect it, or means if we increase its concentration, it increases to this size, this peak. So we can carry out its analysis, and we can carry out the calculations for the pre-concentration factor. We can carry out the back extraction to find out the final concentration of the analyte. So this is the need that pre-concentration is necessary for the suitable analysis of the sample. Similarly, I am showing you a mass spectra, that if you see, these are the small peaks without pre-concentration, and if you carry out pre-concentration, the peak, the analysis, the peak size is increased, and you can carry out, our instrument can detect the unknown analytes, or it can detect the analytes more easily. The response of the instrument is increased. So you can see that these peaks which we cannot analyze, these analytes with these, or these compounds which we cannot see in the sample, after pre-concentration, we can carry out analysis in such a nice way. Because you see, the biological samples, or food, or the soil or sediments, or these samples are very, very complicated samples. They have mixtures of so many things which we cannot, and which we cannot remove very easily. Therefore, the preparation of the sample is very, very important. So this is the need for pre-concentration step before the analysis. It helps. Pre-concentration step is different, and sample preparation step is different, right? Sample preparation step is different. Pre-concentration step is different. Sample preparation, we can, like, we treat the soil sample with water, we treat the soil sample with acid, we treat the soil sample with alkali, and then we carry out, after the sample preparation, we carry out its pre-concentration, and then we subject it to the analysis. So don't confuse sample preparation with pre-concentration step. Both are different steps. Is it clear to all of you, or hello?
Hanji sir, it's clear.
Okay, thank you. Now, if we do not carry out analysis, like here the sample is 80% suppressed, and like after the cleanup procedure, there is a very minimum suppression of the ion. So pre-concentration, or you can say this helps in the removal of the interferences. So we can say that before doing analysis with HPLC, with UPLC, with LCMS, or with atomic absorption, with gas chromatography, mass spectrometry, we must need sample matrix cleanup, that is baseline interferences should be avoided, and there are suppressions in the mass peaks or in the mass spectra, so they should be avoided, and we should carry out pre-concentration. We should apply pre-concentration processing step needed to get sample ready before injecting into the instrument. So pre-concentration, we can say it is the processing step which is required to inject it is a pre-concentration it is the processing step. Processing step means we are making the sample ready so that it is injected into the instrument. So I will come, this was a brief introduction why we need pre-concentration and what is pre-concentration, and what is pre-concentration, and why we need a residue cleanup. So I will give you different pre-concentration techniques. Some I will give you examples of the pre-concentration techniques, and we will discuss some applications of pre-concentration techniques, means with how they are helping us. So pre-concentration technique, mainly that is, these are extraction techniques, and extraction can be a flow through equilibrium and pre-equilibrium, batch equilibrium, and ready state, exhaustive, or it may be a non-exhaustive process. Similarly, flow through equilibrium and or pre-equilibrium, it may be exhaustive or it may be a non-exhaustive. Exhaustive steps include purge and trap, Jorman trap, solid phase extraction, supercritical fluid extraction, and PFE. This is another method for extraction. And similarly, in batch method, exhaustive, non-exhaustive, this is liquid extraction, Soxhlet extraction, and extraction using Jormans and microwave assisted extraction. Non-exhaustive, that is head space, liquid micro extraction, solid phase extraction, and another is steady state exhaustive and non-exhaustive method include membranes. That is, by using membranes we can carry out the extraction or pre-concentration. I will discuss the miniaturized, I will discuss with you miniaturized solid solid phase extraction techniques. This is these are solid phase extraction, solid phase micro extraction, magnetic solid phase extraction technique, MSPE, stir bar job tube extraction, immunoaffinity solid phase extraction, ISPE, micro extraction impact syringe, and dispersive micro solid phase extraction. It is called as DMSPE. We have done a lot of experiments in solid phase extraction or in developing pre-concentration methods. So you see, if there is a no sample blank, how to calculate the recovery or efficiency, extraction efficiency. That is, first we carry out a blank sample matrix. Blank, this is, most of the people don't understand what is the blank. Blank involves everything except the analyte. Blank includes means, suppose you are doing the sample preparation, you are treating with acid, you are treating with alkali, and all other steps, the blank sample should include all the steps, all the chemical residues which you are using in your sample preparation, except the analyte. And with the analyte, then we carry out the analysis with the analyte. So this is that is like we take suppose we take a blank sample, we carry out, prepare the sample, and then add or spike it into it, and then again carry out the analysis. That is, we add the standards into the blank sample. By this way, we can calculate the recovery efficiency, or what is the percentage of our recovery. So this is a response of the extracted sample with analyte divided by a response of the post-extracted spiked sample. Response post-extracted spiked sample. That is, we carry out the analysis without spiking it, and after spiking it. Spiking means we add the standard samples of the analytes, by that we can calculate the percentage recovery. Or there are these 2-3 terms which are involved in pre-concentration. In pre-concentration, first is the enrichment factor. Enrichment factor is like in case of we are dealing with metal, like this is the ratio. And this is Cm over Cn, that is divided by Cm over Cn, that is in the non-polluted sample, major chemical composition, that is baseline versus sample. It is the ratio of these two, that is baseline and sample. What is the peak? And similarly, enhancement factor, that is we are carrying out the analysis, and this is now present in C and D. So we calculate the partition or the enhancement is given by this ratio, that is volume of A, volume of B, volume of C, and volume of D. So by their concentration difference, we can calculate the enhancement factor. Or enhancement factor, simply we can say that it is the final concentration divided by the initial concentration, and by that we can calculate the enrichment factor. Now, why we need pre-concentration? We need pre-concentration. The main aim of the sample, besides the aim of the sample cleanup, analytical scientist, it uses one or another sample preparation technique to have the best possible limit of detection quantification. This means simultaneously bringing the analytes to a small volume relative to the original sample, or onto a solid adsorbent, as in solid phase extraction or solid phase micro extraction, and thus the use of the concentrated analyte for analysis. And the enrichment pre-concentration factor, it is the number of times the analytical signal is enhanced relative to analyte signal without enrichment. The term enrichment or pre-concentration factors are interchangeably by many factors. Enrichment factor or pre-concentration factor, enhancement is used for the purpose, this purpose, as the word this represents signal enhancement. Now, like I will say this, like if this sample, this is your sample without you say peak, without pre-concentration. After this is after pre-concentration, you get such a large response. So this signal divided by this signal into 100, you can find out the percentage of your pre-concentration step. You can find out pre-concentration. So first I will introduce you different sample pre-concentration techniques, and this technique is available in our lab also, and many students, they have done work on SPME. One of your participant, Dr. Gaurav, he has also done a lot of SPME, solid phase micro extraction technique, used solid phase micro extraction technique for the pre-concentration of samples. So I will introduce you and their work. I'm thankful to them. They have done such a huge work, and SPM, so this is known as solid phase micro extraction. Now, let me introduce you what is SPME. SPME is a modified, hello. SPME is a modified syringe-like instrument, and this is a fused silica. It is having fused silica fiber, having small size and cylindrical geometry, is connected to a stainless steel tubing, and silica fiber is coated with a thin film of several polymeric phases, and fiber assembly is reusable and replaceable, and small size and cylindrical geometry uh, of the fiber brings many advantages, such as easy replacement of the absorbent into a sample or head space above the sample or above the sample to extract the analytes. Now, advantages of SPME. There are many advantages of SPME, that it is organic solvent free. We don't need for the for this extraction any organic solvent. It is low cost, though we have used this technique, but I don't agree with this. It is a low cost, but it is highly sensitive and very short extraction time, easy to use, and this does not require any other sample preparation or sample pre-concentration step, and this can be easily automated. And another very important advantage is that it allows field sampling, means sample which are present in the environment, that is anywhere, samples of the analytes from the site, they can be they can be brought to the lab for the analysis, and we can connect it to HPLC, because HPLC offers many advantages. You have an introduction to HPLC yesterday, like it is simple to operate, fast and versatile technique, highly sensitive and good resolution. It is applicable for qualitative as well as quantitative analysis, small size. Small size, wide spectrum of applications applicable to polar, labile, and non-volatile compounds, highly efficiency and more flexible in operation parameters, and reusable columns, and it is safe to use with HPLC. So we have, it is very important that we carry out the pre-concentration step without any solvent. And we carry out the pre-concentration step without any solvent. And another thing is that we can carry out the pre-concentration step in the field. That is field, you take the sampling device, precursor, you take the SPME, and you carry out the pre-concentration step there, and you pack your fiber, bring it to the lab, and then you can carry out the analysis in lab. So how to attach HPLC, how to do the how to combine SPME with HPLC. This step is called hyphenation step. Hyphenation means combining two analytical techniques. That is how we can combine SPME with the HPLC. That is hyphenation. This process means linkage of these two techniques, pre-concentration methods. So this is very important that we carry out hyphenation. So in our lab, we have used Dynex HPLC system, and it is not necessary that it can be done only with Dynex. It can be done with any of the HPLC system. We have used Dynex HPLC system and column reversed with this HPLC pump, and this is a column, and UV detector we have used. We can use it with mass spectrometer also. We can use it with any other refractive index or fluorescence detector also. And SPME, this was supplied by Supelco, and we have attached it with we were successful in performing the experimentation by combining HPLC, by hyphenating HPLC with SPME. So for this we need a rheodine valve, which is about 50 microliter, and SPME desorption chamber, this is 60 microliter. So this is the SPME desorption chamber, and SPME desorption chamber, and we have connected it to the HPLC. And here you can see that main is this is a rheodine valve. To this, the mobile phase from HPLC comes. This is the desorption chamber. Here, the desorption of the analyte from the fiber takes place. That is analytes, and this is the holder of the SPME device. So in this chamber, desorption of the desorption from the mobile phase takes place. So like you can have, and we have like this, it's done in our lab. And this is a HPLC quaternary pump. This is a UV detector. This is the holder. This is the SPME device, and it is attached. It is done in the desorption chamber. That is the desorption of the analyte is taking place. It is all online system that we carry out the pre-concentration step or sample preparation step directly. We don't need any solvent for the solid phase micro extraction by the pump. That is this mobile phase, which is causing desorption in the desorption chamber, which is developed, and this where the desorption takes place, then it is again injected into the HPLC pump and carried to the detector. So mobile phase directly come to the desorption chamber where desorption takes place, and with the then it is injected into the column, and by this rheodine valve, it is connected in rheodine. That column is connected in the rheodine in such a way that after the desorption, it is carried to the column. And then in the column, separation takes place by HPLC, and then it is carried out. Then it is subjected into the detector where the response, UV response, and we can detect the peaks with the help of our software. So this is the hyphenation of SPME. I will more introduce you what this SPME looks like. Like this is an HPLC pump, this is a SPME device, and we have this SPME and column and this desorption chamber. So this is how it looks like. That is, this is a plunger by which we can move this fiber up and down. And if like we want to carry out analysis of this liquid or solid sample, if the sample is liquid or solid, there are two possibilities. This is the fiber, silica fiber, or different fibers they are used. We can modify this fiber. I will give you detailed introduction of the different fibers. Then we can move it or in the sample, we can move, you can move it above the sample or in the sample. This choice is yours, and then that is you can put it in this way, in headspace way, and by this plunger you can move it up and down, and fiber is safe in this holder, and then you can take it anywhere for the analysis. So that is proof. This is, we can with the help of this plunger, जैसे पेन में होता ना, we can make it up and down. So with this help, we can move this fiber up and down. जैसे हमारी पेंसिल होती है ना बेटा, जैसे हमारी पेंसिल को हम मूव करते हैं, we can move it up and down. So this is like a SPME device. It is marketed by Spelco, and this is the lab made device. We prepared this device, and we took a silica capillary, and this silica capillary was peeled off. It was coated with suitable, and this syringe we replaced this syringe we replaced with a simple syringe. So how the coating was done. If we look at this part only, this is the SPME device. Here it is the fused, this is the fused silica fiber on which polymer coating is done. And this polymer tubing, this is a polymer attachment, this is a tubing. In this we can move with the help of we can move with this fiber in this tubing up and down. जैसे हम clutch pencil में करते हैं ना, we can took the carbon inside and outside. This works like that, right? So we can have different coatings on the fiber, like we can have a monolayer coating, we can have a bulk coating, and another is if we use a silica capillary. Silica capillary, you can have, silica is a The silica capillary is flexible. We can modify the inner surface of the capillary, or we can fill any material inside this the inside this silica coated capillary, or that is, we can fill a polymer material inside the capillary, and this will act like a column. This will act like a column. But it is not a column, but under high pressure, or in this gets a modified capillary, and you can see how the this monolayer coating or bulk coating, and depending on your need, you can modify it. And how in the HPLC rheodine valve it looks like. If we want to carry out desorption, that is, like, this is an SPME fiber, this is a pump, this is column. Those who have learned this HPLC system yesterday, we make the connection, that is the mobile phase that comes and fill this capillary and fill this tubing. And here, this is how the desorption takes place. And from the desorption, it is carried into this rheodine valve. And from this rheodine valve, it is carried to the column, where the separation, that is online desorption from the fiber, can be achieved. So using with SPME, you have first step involves the adsorption of the analytes on the SPME fiber, and for this we need selection of extraction mode, selection of the fiber, optimization of the conditions, and desorption of the analytes in the desorption chamber is done. And how to carry out the desorption in a proper way, that is very important. And finally, we carry out the analysis with any HPLC combined with any detector system, like we have done with UV system, or you can carry out with mass spectrometry, or you can carry out with fluorescence detector, or you can carry out with any other detector system. Now, how commercially available fibers are, like, they are made up of PDMS, polydimethyl siloxane. Further coating can be done with divinyl benzene, polyacrylates, carboxane, polydimethyl siloxane, carbovax, templated resin, and stable flex, divinyl benzene, carboxane. All these mixtures, they can be used to modify your choice to make so that the fiber is can very efficiently work. And extraction, it can be done. There are different ways, like simple agitation can be optimizations, how we carry out the analysis. There are two methods for extraction using SPME. First is headspace method, and second is the direct immersion mode. That is, in headspace mode, we keep the fiber just above the analyte. And in direct immersion mode, we dip the fiber into the sample. We dip the fiber into the sample. That is, we expose the fiber into the sample, and otherwise we carry out the headspace mode. That is, in headspace mode, we don't need any in headspace mode, we put the fiber above the sample. In immersion mode, we keep the fiber inside the sample. So if your sample is volatile, you can use headspace mode. If your sample is non-volatile, you cannot use if your sample is not volatile, you cannot use direct headspace mode, then you have to use the immersion mode. So this is again a schematic representation, that is we can carry out how the SPME, this SPME fiber in the desorption chamber, it is connected to the rheodyne wall of the HPLC, and we can carry out the desorption of the sample in the rheodyne. So this is the desorption, and this work, that is, we have applied SPME for the detection of explosives like RDX, HMX, TNT, Teryl, these explosive materials and their metabolites like DNB, nitrobenzene, nitrotoluene, aminotoluene, different isomers, and they can be separated. They were so there is a very good review is available for the analysis of explosives using SPME, HPLC, and other other analytes also for using for other analysis of other samples, like for carrying out analysis for if you want to do UV analysis, then you should know the absorption spectra of these different analytes, and like we have found the absorption lambda max of these these explosive materials like HMX and RDX. We have analyzed and we have studied their absorption spectra, and like you can see, we can use very specifically for maximum for their maximum absorbance in the UV spectra. We can carry out the analysis. Like some of the samples like nitrotoluene has lambda max at 283 and dinitrobenzene absorbs at 238 nanometer. So there is a lot of difference in the selectivity at their wavelength. That is, the signal is the absorption is different in the UV in the UV detector. So it is necessary to find their absorption maxima in the, and we have this HMX and RDX, they are non-volatile. Then we developed an approach for their extraction using explosives, and we found that this HMX, this is an explosive, normally you know, and its extraction efficiency or its peak area is very much increased when we use surfactants. Like we can separate HMX and RDX, like these are their peak areas, and we do this this is without, this is a blank sample, and like this is extraction with SDS. Now I will show you after pre-concentration with pre-concentration with HP with SPME, how we can increase their signal ratio. Like this is the HPLC UV. This is in. These are found in HMX and RDX. They are found in the mobile, in the water, and these HMX and RDX samples, they were detected, and we were able to detect these samples at a very low concentration of 20 ppb. Like this is the extraction is done with SPME in the presence of SDS. Thus, you can see that the sample, the HMX and RDX, which are not visible at all after using SPME extraction, we can detect HMX and RDX. Otherwise, that is this problem is found in somewhere in Chandigarh, like in area where you have a close to Chandigarh, there is in Ramnagar, there are a lot of explosive activities are done by the army, and contamination is there in water of HMX and RDX. They are found. So at different concentrations, that is, you can see the peak height is increasing, and we can carry out the pre-concentration. So with SPME we collected different river water sample, groundwater sample, and then spiked them, and we can say that SPME, that is solid phase micro extraction, can be applied to them. And we published these results in about in 2008, and this technique was developed, that and recovery factors, they were about 100% using SPME. So another thing is that we applied SPME to the analysis of nitroaromatic compounds, which are also explosive materials. And you will see that all these 11 explosives, they can be very easily separated and detected. That is, we can optimize the desorption time, how much time we have to dip the fiber in the mobile phase, and we can carry out the analysis. Like this, we can carry out analysis at such a very low concentration of 20 nanogram per ml. That is, these samples, they were spiked at a concentration of 20 nanogram per ml, and we can carry out pre-concentration of these explosives in water or in industrial samples at such a low concentration. And you can see that the detection limits are very, very low for the. So we can say that SPME, it can be applied to air samples, it can be applied to water samples, it can be applied to soil and sediment samples, and these are the different literature applications that that is like if you have an organic pollutant, it is present indoor air sampling, then we can use PDMS fiber, and GCMS can be used. If you have a BTEX benzene, toluene, ethyl benzene, and xylene and hexane, then air sampling can be done, volatile organic compounds. So all these samples using a suitable fiber, you can carry out with a you can carry out analysis with a suitable instrument. Like with metals, you can carry out with GCMS or ICPMS and BTEC. No, all these that is many applications of SPME are done in environmental and soil samples. So much useful this technique for the direct analysis of for the direct pre-concentration of the environmental pollutants in the so we can conclude that SPME is a solvent-free micro-extraction technique. It is cost-efficient sample. It is very simple and in use, very highly sensitive, low detection limits can be attained, and it can be used to sample analytes of many types, and it can be used in industry. Another extraction technique, micro extraction technique, pre-concentration technique is micro extraction by packed absorbent. Micro extraction by packed absorbent. This is known as MAPS. MAPS, MEPS, micro extraction packed absorbent. M stands for micro extraction. M stands for micro, E for extraction, P for packed, packed absorbents. So in this, this is a packing material which is filled. This is a bin, and in this packing material, it is placed in this, packed in this syringe, and by this plunger we can move the solvent in and out. So we put this packing material into this. Hello. We put this packing material into this syringe, and with the help of this needle, through this needle, this can be pierced, this can be inserted into any sample, and by that repeated cycles, we can make the pre-concentration steps. And again, it is a miniaturization of the conventional solid phase extraction method. SPE absorbents, they are packed in the MAPS bean, and syringe can be attached to gas chromatography or liquid chromatography, and MAPS is very suitable to reverse phase, normal phase, mixed mode or ion exchange chemist. That is, we can use different type of materials, and MAPS is also currently available in different SPE phases, and packing can be done. And the main advantages are it reduces the time to prepare and inject samples from how to prepare the sample step from hours to minute. It reduces the sample volume, which is we need only about 3.6 microliter of the sample, and it reduces buffer and solvent, which are very less, and all extra steps they are avoided. That is between or after the sample preparation, they are avoided. So these are the different advantages, like it reduces sample volume, it can be fully automated, it reduces buffer and solve time for the preparation of the sample is reduced from hours to minutes. So we can connect maps with that is in the micro extraction in the packed syringes to the gas chromatography. Like this is our sample, we can take the sample in, we can take the sample out, means we can, by this cycling step, we can wash it again, we can elute the sample, and then we can inject the sample either into the GC or into the HPLC. That is either into GC or HPLC, whatever may be the detector, that is UV detector, fluorescence mask. Similarly with gas chromatography, whatever may be the detector, we can connect it directly to the detector. So GCMS system, generally we have done the analysis with GCMS for the analysis of explosives. Like GCMS system, it consists of RTX column, and that is different standard GCMS was used for the processing. So GCMS system, that is, it consists of like yesterday you had a very good discussion with the GCMS system, and so we can analyze the mass spectra. Just a minute. So in gas chromatography, you have carrier gas. These are the conditions which we have used. We can, and in the application part, these are the MS conditions. Like we can have a temperature of 290 degrees centigrade, and electroionization source was done at 17 electron volts, and solvent cut time and mass spectra were recorded. So before mass analysis, like these are the different fragmentation pattern for different explosive materials, nitro explosives we have carried out, and we have prepared their calibration curves, and we have optimized all the conditions for the explosive materials like nitrobenzene, nitrotoluene, and different tetrail, all these explosive materials, uh, their extraction conditions, they were optimized with different solvents and with the different solvent volumes, all these, and optimized the pH. So finally we were able to, that is, you can see the GCMS. This is the in the UV spectra using HP maps GCMS chromatogram for the under the optimized conditions, and then we have applied this for the analysis in blank when different tap water or river water or different uh, samples, they were collected and conditions were optimized for the analysis of these explosive materials. And you can see like RDX and TNT, they can also be analyzed. And in blood samples, in groundwater, and these are the different publications you can see and you can get from us for the analysis of these explosive materials, or from the or other materials also we have done. And another technique is the stair bar adaptive extraction technique, and this is a stair bar adaptive extraction SBSE. And this can be applied to a wide range of samples. That is, this stair bar is coated with a different polymeric material. And again, like this can be done with headspace mode, or it can be done in a by immersion mode, that is direct in immersion or in headspace mode. If you can hold it with a fiber, or you can with a sport, or you can dip it in the solution, and analytes, they are adsorbed on this polymeric material, and you can analyze fruit, food, drug, environmental samples. All advantages, they can be obtained. So I will show you how it works. That is, this is a glass material on which PDMS coating is done. Inside you have a magnetic, inside you have a, you can say you have a iron on iron rod or magnetic core, which can cause you to, it can move in the sample. Adsorption can take, like if you put it in water, you put a stair bar into it, extraction is done, and then you put the solvent in it, desorption will take place in that small analyte, and desorption will take place in that solvent, and the sample which is prepared, you can inject and you can carry out the analysis. That is this bottle, which is having your any drug, pesticide, explosive, any environmental contaminant, that is that these analytes, they are adsorbed on this they are adsorbed on this PDMS coating, and then they are desorbed. That is from this large volume to they are carried to this much small volume. That is pre-concentration, right? And this is chromatography can be done, and all these analytes they can be analyzed. So here I have selected a latest reference like development. You can see I will discuss this publication and how it is done. That is my main aim is how you can apply stair bar SPSC, stair bar adsorptive extraction, to the analysis of water and urine samples. And for if you want to see the details, you can go to this reference, or you can download this paper. And first step is sample preparation, like they have collected the water samples from the river, from the tap, and they have stored it at 4 degrees centigrade. And after that, all these urine samples, they were used as such without any further treatment. So SB stair bar adsorptive extraction was how it was applied. That is stair bar adsorptive extraction units. They were conditioned with water. First you washed it with water for 5 minutes. Then stair bar was placed in 15 ml of the sample and stirred for 30 minutes. And after that, this was analytes were adsorbed on it, and then they were removed from it by methyl alcohol. And subsequently, then you take 20 microliter of the sample and inject it into the HPLC or GC system. They have shown that how this coating, that is, they have prepared some monolith materials and metal organic frameworks using monolith, and this material, that is the surface of the stair bar, how it looks like, and this is a SAM image, and on which you can carry out the adsorption studies. So another how you can carry out the coating, that is you take the MOF material and you add to it this you take the MOF material and put the stirrer in it, and then you stir it for about with the polymer and reflux system, and you heat it about for 48 hours. There is MOF and monolith, they can be put on the, they can be adsorbed on the stair bar. So you can see the application of different samples, like in tap water, in human urine, tap water, and in river water. That is how nicely that these analysis can be done with the help of stair bar adsorptive extraction. Another method is magnetic stress, magnetic, you can say magnetic solid phase extraction. This is also very interesting and very easy to carry out. And in this magnetic solid phase extraction method, this is also, we like, this is the sample solution. We add to this magnetic nanoparticles to it and stir the solution for it, so that the analytes, they get adsorbed on the nanoparticles. Adsorption takes place, and then we collect, we separate these nanoparticles which are magnetic and adsorbed with the analytes with the help of a magnet, and drain it out, solvent, and then put some solvent, then carry out the redissolve it in a small volume, take the sample for analysis, and carry out the analysis. So this magnetic solid phase extraction is applicable only to the nanoparticles, or only to the stir bar adsorptive extraction that is magnetic in nature, and rest remains the same. That is, we put some magnetic material, or we can put some nanoparticles which are magnetic in nature, because nanoparticles have more surface area, more adsorption of the analytes takes place on the nanoparticle surface, and with the help of magnet, these nanoparticles, they can be separated, on which adsorption has taken place, and then we can carry out the elution, and after the elution, this desorption can be done, and separation can be achieved. Separation can be achieved. So another is immunoaffinity solid phase extraction. And for example, you can have a reference of it, Journal of Agricultural and Food Chemistry. There is an example in immunoaffinity solid phase extraction. Like you see, there is a immobilized resin which is very sensitive for small molecules, protein, or antibodies, and these with this coating which is on the resin, so this can attract and this can pre-concentrate small molecules of proteins or antibodies, and non-specific molecules, they can be removed. So another important extraction method is single drop micro extraction. Single drop micro extraction, again, this is very useful, and you will be wondering how, instead of a large solvent, we can carry out analysis in a single drop. And a single drop analysis is done like this. We take this micro syringe, which is having this needle, and it is a septum. We, with the help of this plunger, we make a drop of solvent inside on the tip of this micro syringe. And if this organic drop is not soluble in this solvent, we can carry out if this is not soluble in this solvent, we can carry out direct immersion. And if it is soluble in this solution, or in this solvent, in this solution, then we can carry out head space single drop micro extraction. It is so simple, and that we need only a small drop of organic liquid. And these, either you heat it, if your sample is volatile, they will be dissolved in this drop. And if you can carry out on this, you stir the sample, organic liquid drop is insoluble in this solvent, and this will extract all the analyte which is present in this sample. So this is headspace or direct single drop micro extraction can be done. So we can, I have shown you that is sometimes this is this technique is modified and in different ways, like in direct immersion I told you, and we can make a above the organic liquid we can put some another solvent in which this drop is stable, that is called three-phase single drop micro extraction, or we can put it inside the tubing, or we can put it like a bubble. So these are the different modifications of the solid phase single drop micro extraction, and this is in the GCMS, or how we can with the help of a micro pipette we can carry out a single drop micro extraction. So analysis, how it is done. Another extraction mode is membrane. This is MSB, liquid phase magnetic solid bead, or liquid phase micro extraction, and it is attached to gas chromatography. And again the same principle, that is, if we put a stainless steel wire in it, it keeps on moving in a solvent in the sample, and where and with the help of a magnet we can we do not allow the sample to be eluted out. Then with the help of an organic solvent we can carry out desorption, and with the help of desorption we can carry out the elution, and with the help of nitrogen we can pre-concentrate it further, and because nitrogen gas helps in the evaporation of the solvent, and we can carry out analysis using gas chromatography. So this has been applied, and different modes of this micro extraction technique are possible. That is, you can put in small pipette, or you can put you can put a silica tubing, and on which this extraction or membrane movement of the analyte can take place, and so analysis can be done. Another method is solidification of the floating organic droplet in dispersive liquid micro extraction. In this mode, what is done, you add an organic solvent into the liquid, and this this extracting solvent gets solidified. That is dispersive liquid micro extraction, solid floating organic droplet is formed, and in this case like this, in this case you take, this is your sample mixture containing solvent, and this you add organic dispense, you add organic solvent and a small amount of the solvent, or which can be solidified. And as it will come through this sample, all the sample, all the analytes, they are adsorbed on this solid surface. And or another thing is, you can solidify it with the help, if it is not soluble, uh, you can solidify it with the help of an ice, and you can remove the organic phase, and you can dissolve it, and you can carry out the analysis. Another is this is dispersive liquid micro extraction. This is another extraction technique developed in the extraction or pre-concentration technique, and it is developed in the last decade. And in this case, like we take this tube, we take this tube and we inject into it with the help of a syringe. We inject into it a solvent or solvent mixture, and this solvent mixture in the form of bubbles, when it comes, it forms another layer. We centrifuge it, and we can decant it off, or we can remove it, and after the removal, that is, we can remove the organic solvent by nitrogen, or we can pre-concentrate it. So different liquid membrane extraction technique, these are developed, and you can see like iron or this membrane and this liquid extraction and this dispersive liquid extraction that can be done. So this dispersive, this I have shown you. That another thing is that we can directly extract this solvent, and we can carry out its analysis. So we can have a comparison of these techniques, that is solid phase extraction and SPME, stair-bar adsorptive extraction, and MAPS. These are the common extracting techniques, and the lowest small volume used is in that is in SPME. We need a small volume, but these some advantages and disadvantages are there. So best is you can say, still we consider that solid phase extraction is considered as a most suitable technique for the pre-concentration step. Another, this is the last, and I will take your few minutes more, molecularly imprinted polymers. They are widely employed for the sample preparation, pre-concentration, and analysis. Now, molecularly imprinted polymers, they are the synthetic polymers, and we can increase the selectivity for a given analyte or a group, and for a structurally related compounds, making them ideal materials to be used in separation processes. That is, we use different templates for different applications, and we can use different molecularly imprinted polymers for the analysis. Like this different, we provide a template. Template means the substance for which we want to make the polymer, for which we want to make the molecularly imprinted polymer, and these are the monomers. We carry out the polymer in it, polymerization in the presence of the template. These cross linkers and these other things, they polymerize, and uh, a space or cavity is created, and this space or cavity can be utilized only for this template. This is known as template, and we can easily remove the template, and this can again rebind it. So depending on this molecularly imprinted polymer, MIP, solid phase extraction, MIP, magnetic or dispersive, molecularly imprinted, and for the sensors, these molecularly imprinted polymers can be employed more clearly. Like these are the monomer units, they are polymerized together in the presence of this template, and then we remove this template, and this imprinted material is created. And this type of the molecule, wherever it is present, that can be fitted into it. And we have prepared imprinted materials, molecularly imprinted polymers for carbamazepine, carbazepine, primidone, for many other materials. Like we take carbamazepine, and these are the polymerizing materials. This in the monomer, this carbamazepine is trapped, and then we remove it, and these vacant space or this molecularly imprinted polymer of carbamazepine, it is not having carbamazepine, that is carbamazepine imprinted polymer is created. So we have applied it for the analysis of carbamazepine and oxycarbamazepine, and you can see we can reach a detection limit of 0.488 nanogram per ml, and oxycarbamazepine 0.5. You can see that at such a low level, the analysis can be done. And if you want to see how we can make carbamazepine, we can make MIP, molecularly imprinted polymers. We have prepared carbamazepine imprinted polymers, and applied for the analysis of carbamazepine in water, like drinking water, river water, wastewater, various pharmaceutical samples. And for this, we have prepared porous molecularly imprinted solid phase extraction materials, and they are suitable for the analysis of carbamazepine at a picogram per level. So you see, this is the sum of the molecularly imprinted material. Like these cavities are formed. This work was done by Irshad and his postdoctoral fellow in Chandigarh. And that is these cavities you see at different resolutions of stem. These, how nice these cavities are. And some image, it appears, and you can directly say that, and carbamazepine, whatever may be the sample, that will be trapped in this template which is created for it, and it can be applied. So similarly, we have prepared porous molecularly imprinted polymers for diclofenac and diclofenac, you can see again that is these cavities. These are the molecularly imprinted cavities for among the monomer units. That is, we take the carbamazepine and different monomers, polymerize it, and remove the we take diclofenac and polymers, we carry out the polymer polymerization, then remove the diclofenac from the polymer, and you will get such cavities. This is experimentally proved, and these cavities, diclofenac molecules, whether they are present in urine sample, they are present in any water sample, or they can be trapped, because these cavities are highly specific for diclofenac. They strongly capture the diclofenac molecule very, they capture the strong diclofenac molecule, and you can see that we have applied them to the sewage sample, sewage treatment plants, and so this was all uh, about the different techniques, different and if you have any question, I will be happy to answer. Thank you very much for your kind attention. Thank you very much.
Thank you very much, Dr. Malik, for your such an elaborative and nice talk on this pre-concentration method. It has covered almost all the techniques. So if there is any query from any participant or any feedback from any participant, please unmute yourself. We will be having a separate session on solid phase extraction techniques, because it itself is a very happening technique in pre-concentration methods. So if any person is having any query,
Any suggestions for the improvement, because we don't want, if you don't want any, ask any question, please give us your comments or for the suggestion or improvement. Everything is welcome. Any criticism, anything, don't hesitate to ask anything.
so there seems no response or no query from the participants.
Are you there or disappeared all?
They are very much here. They are posting the comments in the chat box. They are thanking you.
Thank you, sir.
Somehow they are not, willing to talk.
Thank you, sir. It was really very informative lecture and an elaborate one.
This is a great response, Dr. Kuldeep. Putting in the chat box, that is a one thing, but if we unmute ourselves and just feel these words, then these are more encouraging words, and it also gives impression, yes, everyone is attentive.
Yes, sir. I think on this platform there should be a response, and we need to be little interactive as well. So sir has put in so much of effort for the last 2 hours, and I think we should all thank him for that.
Yeah, definitely deserves applause. Thank you so much. Thank you, Dr. Malik.
Thank you, sir. So we can continue with the next lecture.
Yes. So the next session is by me only, and it will be a tutorial on mass spectrometry. And so in the last 2 days, we had discussed a lot about mass spectrometry, the basic concepts, the instrumentation, the theoretical concepts, then the recent advances. Dr. Yolanda has discussed very well, and some instrumental aspects of those techniques also. So here uh, in this tutorial, we will be, discussing about some factors which need to be taken care of while developing LCMS or GCMS method. Because this is a highly sophisticated technique, a little variation, a little change, a little fluctuation from the optimized conditions leads to highly undesirable results, and that means the loss of energy, loss of man hours, and of course the time loss and economic loss. So just like see, developing a method, analytical method is like building a home, building a house. So like we plan everything before we build a house. Similarly, we plan everything before we start developing a method. See what are our target analytes, what are our purpose, means whether it's a qualitative or quantitative, then what is the expected concentrations, whether it is in a micrograms per ml or picograms per ml or nanograms per ml. Uh, so these all and the resources available with us, these all factors, they are first considered into, they are first taken into mind before we start developing a method. Then based upon this input, we uh, go through the literature, we go through the reports which are already available related to those particular analytes, and we derive a strategy that is so that it is conducive to development of a method in the minimum possible time with the minimum possible number of trials. So what are those factors which are governing the success of LCMS method and how to optimize it? That we will be discussing in this session. So just allow me 2 or 3 minutes to share my presentation. So is the screen visible to all?
Yes, sir.
Okay. So coming to the tutorial on the mass spectrometric analysis of contaminants. So again, network issue. Just let me resolve this network issue. It will not be uh, uh, detected. So first factor, it has to be ionized. Now there can be certain components in the sample or in the sample solution which we have prepared for analysis, which affect the ionization. So those components may suppress the ionization. They usually suppress the ionization, only some very rare circumstances, so they enhance the ionization. So ionization of any analyte, it is prone to disturbance from some components of the sample. And this is a particularly prominent or observed in the biological samples. Biological samples means the samples which are withdrawn from animal sources or from the humans. Now, if these components, how they can interfere, now if these components, they co-allute with the analyte of interest, then data can become biased or have poor decision. So it is possible that there can be some components in the matrix or in the sample which are co-eluting with the drug which we want to, or the compound which we want to analyze. So if they're co-eluting, means they are overlapping over each other in the same peak, right? Then our interpretation will be wrong, or it will be biased. So this matrix effect, it is also affected by the viscosity effect. How? In this matrix effect is particularly seen in case of ESI, electrospray ionization. It is not prominent in atmospheric pressure chemical ionization or chemical ionization or atmospheric pressure photoionization, because in those cases, in APCI and APPI, the substances, they are already in the gas phase. But in case of ESI, the substance which we are going to analyze is in the liquid phase, and in liquid phase, there will be the requirement is that the liquid is converted first into vapors, and those vapors are then dissolved to convert into dry mist. Now the rate of dissolution is very important when it comes to ionization in electrospray technique. Now, if there are some components which are present in the matrix, those components, they will make the sample viscous, they can change the viscosity, and that viscosity will change the atomization process or nebulization process, means conversion of the stream into vapors and subsequently dissolution. So any change in viscosity can lead to significant changes in the ionization. So if the molecule, the samples, they are not nebulized properly, they are not dissolved properly, they will not be ionized properly also. Now, how to assess that there are matrix effects? First thing first, that we have to know whether there are matrix effects or not. Now, there are two ways by which we can do it. One is the post-column infusion method, and second is the post-extraction spike method. Now, in post-column infusion method, what we do is that you are seeing that there is a LC pump which is feeding some mobile phase to column, and the column's element is going to the mass spectrometer. And between column and mass spectrometer, you are seeing an infusion pump. Now, this infusion pump is delivering a fixed concentration of the analyte which we have targeted for analysis in mass spectrometer into the mass spectrometer. So what we do first, before we start the sample analysis, what we do, we have taken the pure solvent. Let's say we have taken water, right? We inject water from the injection port and run the mobile phase, right? It will reach the mass spectrometer. So a baseline will be created. Now on that baseline, then we start infusing the analyte from the infusion pump. So in this case, only our analyte will be entering into the mass spectrometer along with the water. And mass spectrometer will create a response. It will give you a peak in the response. Now that peak has a certain characteristics: area under the curve, it's a peak width, it's a peak height, right? It's a retention time. So these are the various characteristics of that peak. Now, while the analyte is being infused, now from the injector port, we inject the blank sample. Blank sample means the sample which we have targeted to analyze, right? In what form it is. Now that matrix, that sample blank will get mixed with the analyte, right? And it will reach the mass spectrometer. So if there is any matrix effect, the response of that peak, the characteristics of that peak will change. If there is ion suppression because of the matrix, then the peak height will decrease, area under the curve will decrease, the width may change, right? So this way we come to know that matrix is affecting the suppression or ionization of the analyte. If there is no change, then we say that there is no matrix effect, or the sample components, they are not affecting the sample or analyte. So this is the one way. And the second way is post-extraction spike method. Means, let's say we want to analyze some contaminants in blood. Now, blood can be treated in two ways. One is as a serum or as plasma. Let's say we have taken plasma. Now, in plasma, there are several proteins, and we have to extract the drug from that plasma, right? So what we do, we process that plasma sample with suitable extraction techniques. That may be protein precipitation technique, that may be liquid extraction techniques, that may be solid phase extraction techniques, that can be or any other pre-concentration techniques that Dr. Malik has just discussed. So using those techniques, we extract the drug from the plasma. Now, if we want to see how much ion suppression is there, then what we do, we spike the blank plasma or the known sample plasma with a known concentration of the same drug. Let's say we are analyzing the concentration of, for example, any cyclophosphamide in the blood. Now, we know there is some concentration of cyclophosphamide present. Now, what we do, we spike a known concentration of cyclophosphamide, for example, 100 microgram per ml into that, and we treat it with a certain method which we have targeted for this extraction, and after the extraction we feed it into the sample, into the system, and we repeat the same process with the non-interacting matrix like water. So we spike the same concentration of cyclophosphamide in water, and then we treat it in the same way the plasma is treated, and we inject it. Now we have two samples. One is without any matrix, another is with matrix. So we compare the two results. If there is any you see suppression in the, or any decrease in the response of cyclophosphamide in the plasma sample, it means some ion suppression is being done by the plasma proteins or by some components in plasma. So this way we can come to know whether some ion suppression is happening or not. Now, either of the two techniques, if we use them in isolation or separately, may not give you the required results, or may not give you the required information or a reliable information. So for that purpose, we use these two techniques simultaneously. We also go for the spiking studies, we call them the recovery studies, and we also go for the post-column infusion method, so that we can come to know whether there is any ion suppression or not. So this is some practical example which is shown here. So see the plasma concentration of different drugs in different vehicles. Vehicles means the matrix in which there can be some interference or some ion suppression. In this graph, see here, there are the two graphs. One is the diluted samples with APCI mode, and the second one is undiluted sample with the, this is electrospray ionization mode. Now, this is the top one is the diluted sample with APCI, and this is the undiluted sample with the electrospray. So as you can see here, that the area under the curve, or the not area under the curve, the concentrations at different times is this is a pharmacokinetic studies, right? Performed at different time intervals, collected blood collected at the different time intervals. In undiluted sample, means the concentrated sample, the concentration is lower. The concentrated sample concentration is lower, but in diluted sample the concentration is higher, which is the reverse one. Actually, the concentration in the undiluted sample should be higher, but here it is the opposite one. So from here, there is one thing that which is clear, that ESI mode is more affected by the ion suppression or a matrix effect, but APCI is not affected. So we have taken APCI as a standard or as a lead, right, or as a reference. Then we see here in the second graph, this is again the large one, this inverted triangle. This is undiluted sample with the APCI, right? And here the circular one, which is overlapping the APCI graph. It is undiluted sample in this electrospray ionization, and this one, square one, this is the undiluted sample in electrospray ionization. And these the difference between these two is the vehicle. The vehicle is different, right? So the drug is same, but the vehicle is different. So one vehicle is exerting matrix effect, this one, and the another vehicle is not exerting any matrix effect. So we also come to know what should be the diluent for analysis, whether it is a for LC-MS analysis. So this way we select the diluent, and see from the table here again the uh, same data. The drug A, which is shown here, this is for drug A, this is for drug B, and area under the curve, as you can also see, area under the curve for APCI mode with the 5-term diluted is higher than the area of a drug A with undiluted sample in ESI mode. So these studies, they indicate that matrix effect or ion suppression is affected in two ways. One is there are certain vehicles or solvents which cause ion suppression, and there are certain techniques which are more prone to ion suppression, and among that it is a ESI mode. And as yesterday we talked about that, about 80% or 90% of the LCMS experiments are performed in ESI mode. So it becomes very pertinent to pay special attention to detect if there is any matrix effect or not. Now, how we can reduce this matrix effect? How we can minimize the ion suppression? We cannot completely remove the matrix effect, but we can minimize. First thing is, the simplest technique is dilution. If we dilute the sample with the non ion suppressing vehicle, then it will prevent the ion suppression, and the concentration area ratio right will be optimum. Then second is the lower flow rates. Now in ESI mode, as we have already discussed, that first step is a nebulization and then it is a desolvation. Now if the flow rate is low, then in the chamber there will be sufficient time for conversion of, or there is you see good efficiency in the conversion of the liquid phase to vapor phase, and from vapor phase to gas phase, right? If we increase the flow rate, then the temperature, we have to increase the temperature in the chamber. To promote that, we have to increase the gas flow also to compensate that increase in the flow rate. And so that nebulization may not be effective, may not be complete, desolvation may not be complete, so that will result in incomplete ionization, therefore poor response. Third approach is the chromatographic. So we can physically separate the analyte and interference on columns so they elute at different times. So it is possible that the analyte, the component which is causing interference, it can be eliminated or it can be resolved, it can be separated on HPLC or through the chromatography technique, whether it is a GC or HPLC. So if they are, so that they elute at different times and they do not interact with each other. Fourth is extraction. We can selectively remove the interference during the sample preparation stage, like solid phase micro extraction, like liquid extraction, like protein precipitation. Like in biological sample, usually we go for protein precipitations. Why proteins are required to be precipitated? Because proteins are highly polar in nature in comparison to the organic compounds which we are going to analyze. Now those polar organic compounds, the proteins, right, they will interact with the substance and they will suppress its ionization. So its ionization to increase the ionization of that compound, we have to remove those proteins. So there are the different ways. One is one that we call it a protein precipitation. So we can use some acidic environment, like trichloroacetic acid is very frequently used. We can also use methanol or acetonitrile as a protein precipitating agent. So we mix those plasma with the precipitating agent and we keep it for a few minutes. All the proteins, they will get precipitated. You centrifuge it, and after centrifugation you get the supernatant, and that supernatant is usually either free from the proteins or they have the very less concentration of proteins. So this way we can extract the components, required components from the sample, so that interference is removed. Last part is the compensation. Compensation for the ion suppression. So it involves the use of a stable isotopically labeled internal standard or matrix match, so that any effect is consistent. So the elution time of this isotopically labeled internal standard will be virtually identical to that of its unlabeled counterpart and will therefore undergo same amount of matrix effect. So this is, you see, similar to use of recovery studies as well as use of internal standards. So that's why all the biological matrices, when we analyze, let's say, brain sample, we analyze liver sample, we analyze blood, we analyze cerebrospinal fluid, any type of biological sample is used, analyzed, it is mandatory to add internal standard into this. And that internal standard should have properties very similar to the properties of analyte which we are going to analyze. Means its a pKa value should be very similar, its a log p value should be very similar, its a molecular mass should be very close. So these are certain requirements which we consider while selecting the internal standard, so that if there is any ion suppression, so that ion suppression can be compensated. So how we compensate it? We take the blank sample of let's say we want to analyze the component content in plasma of drug A. Now plasma is spiked with the internal standard first, and it is subjected to the extraction process, which is a preset, and it is analyzed on the LCMS. And so LCMS will see whether there is, what is the ionization of that particular internal standard. Now then we carry out the spiking of internal standard in the plasma containing the drug A, and again set the procedure. So if there is any interaction, that will be nullified by the previous chromatogram which we have obtained. So there will be overlapping, there will be noise reduction, there will be background reduction. So that will cause us compensation of the contribution by the internal standard or by the matrix. Okay, the second factor which affects the optimum use of the optimum performance of LCMS is the nebulization process. Nebulization is the very first step in mass spectrometric analysis. So the ion production may be maximized. So the sensitivity of any LCMS method lies in its sensitivity. It's a concentration of or abundance of the ions which are produced from the sample, and this ion production may be maximized, and therefore the instrument sensitivity can be enhanced by optimizing the two things, that is a droplet radius and the number of charge on the droplets. So droplet radius has to be decreased. Lesser is the droplet radius, more will be the number of charge on the droplets, better will be the desolvation, and better will be the charge distribution. So formation of a stable nebulization aerosol is dependent upon several factors, and the first factor is the potential difference between the electrospray capillary and a counter electrode. So yeah, this is potential which is applied across the capillary, right? And there is a counter electrode, means how much potential is applied to this, so that gradient will increase the polarization of the material. Then second is the nebulizer gas flow. So this is the sample coming from your LC part, and this is the outer capillary nitrogen gas is fed into this. So what is the nebulizer gas flow? So to convert it into an optimum stream of vapors, right? It has to be the nitrogen gas flow has to be optimum. If the gas flow is very low, it will not be converted to vapors. If it is a very high, it will be again not converted into vapors. It will cause a splattering of the liquid, right? So you get bigger drops. Say, and then third point is the spray position relative to the sampling orifice. Sprayer position relative to the sampling orifice. So this is uh, spray position here, right? So this is the how much orifice is there, this one. So because it affects the, you see, direction and the speed of the gas, and the eluent composition and the flow rate. So what is the flow rate of this eluent which is coming from the HPLC or UPLC or GC, and what is the composition of that mobile phase? If a mobile phase is viscous, then nebulization process, to make a nebulization process effective, we have to increase the flow rate of gas. If if the element is not viscous, it is a less viscous, then we need a lower rate of gas flow. So these are the various factors which affect the nebulization process. So here is the effect of the gas pressure or the gas flow, or we can say effect of nebulization on the ionization. See here, this is some protein sample which is analyzed. This is, sorry, cytochrome. This is cytochrome C, which is analyzed at different gas pressures. So from 0 to 50. So as the gas pressure is increased from 0 to 50, see the intensity of the ions. Intensity of the ion plus 6 and 7,6 means the cytochrome C is protonated at 6 sites, and plus 7 means it is protonated at 7 sites, right? And this is you see an LCMS total ion chromatogram of a cytochrome C recorded when the pressure is 5 bar here and the flow rate is 5 microliter per minute. So this chromatogram is corresponding to this point, right? So here, as expected, so 6 times protonation, and this is 7 times protonation. So obviously the M by Z with 7 times protonation will be less than the M by Z with 6 plus protonation, or the 6 times protonation. So as the gas pressure is increasing, ionization is increasing beyond a certain value, like up to 20 here for the at when the flow rate, when the this is this one, bold line is plus 6 and the dotted line is plus 7. So for both 6 and 7 at this 5 ml flow rate, when the flow rate is 5 ml, the ionization is maximum when the gas is flowing at gas pressure is 20 bar, and beyond that its ionization decreases because vaporization is because nebulization is not effective, right? And for if we increase the flow rate, if we increase the flow rate of the liquid entering into the detector, see there is a continuous increase in the intensity with the increase in the pressure, and this is tested up to this 45 bar. It was not done beyond 45 because that poses another hazards of explosion or so on. So as the liquid flow will increase, the gas pressure has to increase the ionization, right? And and for every flow rate, there is an optimum pressure for maximum ionization. So this is see a simulation of the gas velocity profiles for the same experiment at various gas pressures. This is a computer simulation where we can see when the gas pressure is 5 bars, 10 bars, 20 bars, and 45 bars, right? So as the pressure is increasing, then this cone size is increasing. Cone size is increasing. And this is a detailed description of gas velocity, right? Map with a nebulizer gas of 45 bar pressure. So this is your capillary, this is your from gas coming here, and up to see certain distance, it is stream, and then it is converted to spray, right? And this is what is happening in the here near the exit point. So stream is there, then it is converted to drops, right? It starts converted to droppings. Okay, the next step is desolvation process. Once the nebulization is done, then we have to dissolve the droplets so that the solvent is removed and the spray is converted to dry mist, and the ions are individually ionized. The molecules are individually ionized. Now, the most effective desolvation is achieved by a combination of processes and the parameters for which are interrelated and should not be optimized in isolation. Means there are certain parameters which means like a desolvation process. If we simply increase the temperature of the chamber, right, it doesn't mean that the desolvation process will increase. Desolvation process will be efficient, right? So there are various factors which we have to consider. Again, one is the what is the source temperature, means what is the temperature of that chamber. What is the temperature of the liquid which is entering into the chamber, and second is the drying temperature or desolvation temperature and the gas flow. So what is the gas flow and what is the desolvation, what is the temperature of that chamber, and second is the declustering, how effective the declustering is. Declustering is explained here. See, this is again a molecular dynamic study which depicts the how a molecule, how a sample is nebulized, it is dissolved, and how the number of molecules they change. So this number in this number or the top indicates 50 picoseconds, 715 picoseconds, and 1200 picoseconds. So within you see around 1150 picoseconds, the whole dissolution takes place. Almost complete dissolution takes place, right? So such is a fast process. And if we concentrate here, so see here, this is your molecules, your analyte, which are clustered together, right? And this white color, they are the water molecules, right? So as we move from left to right, see the number of water molecules, they are getting evaporated, they are getting dissolved from the substrate. So here our substrate is having some water molecules. Here the water molecules from the substrate are released, so means it is getting compact. So this is a larger size, this is a compact size, and this is here still compact size, and it's a very lesser number of water molecules are seen. So desolvation has taken place. And here after 1200 picoseconds, see here you are not seeing any cluster. You are not seeing any cluster of the analyte of the substance. You're seeing individual molecules here, red one, here red one, here red one, here red one, this red one. So this is how the molecules, your substance, it is declustered. So the declustering is different for every kind of analyte. So the three factors: what is the source temperature, what is the drying temperature and gas flow, and what is the declustering behavior. So they totally affect the desolvation process. Now, desolvation is also important in certain kind of experiments, which nowadays we perform hydrogen deuterium exchange experiments in mass spectrometry. What is the importance of these experiments? Hydrogen deuterium exchange experiments, they are performed to come to know how many number of exchangeable protons are there. How many number of what is the number of exchangeable protons are there? For that matter, let's say if we have taken this example of hydroxybenzoic acid, this is 2,4-dihydroxybenzoic acid. So here the exchangeable protons are, this is a 1 benzoate, this is a hydroxy, this is hydroxy. So they are the exchangeable protons. So under normal conditions, these will be exchanged. So if they are exchanged, their molecular mass will increase. So the basis of a HD exchange experiment is that if we exchange, so how this is exchanged, we prepare the sample solution and mixed it with some drops of heavy water, deuterium oxide, and keep it for some time. And whatever the hydrogens are replaceable, those will be replaced by deuterium, and then it is analyzed. Deuterium is having a mass 2, whereas hydrogen has a mass 1, so they will be detected at a 1 mass unit higher, right, than this. So see here, if this is a 153 is a molecular mass of this particular ion. So this is a ionized in a negative ionization mode, right? So molecular mass of a this dihydroxybenzoic acid is 153. So you're getting this peak. Now, if you perform this desolvation at 200 degrees Celsius, then you will see two peaks, one at 154, another at 155. Means at 200 degrees Celsius, either there are two possibilities: either the one of the two hydroxy groups, they are exchanged with the OD, means hydrogen in this or this is replaced by deuterium, or both can be replaced by deuterium. So if one is replaced, the mass will be 154. If two are replaced, then the mass is 155. But if we carry out the desolvation at 400 degrees Celsius, then what will happen? These two hydrogens, they also get displaced by deuterium. So when they are displaced, their mass will increase. So presence of these peaks in the mass spectrum indicates that at 400 degrees Celsius, even the non-replaceable hydrogens are also exchanged with the deuterium. So that is causing huge. 400 degrees Celsius means giving such a high amount of energy that even the normal carbon hydrogen covalent bond is broken, and it has undergone exchange reaction with that. So this is the importance of desolvation temperature. Means we cannot simply increase the desolvation temperature to ensure that yes, the ionization will be good, because excessive desolvation temperature can lead to some other intermolecular reactions, so that alters the nature of the molecule which we are going to analyze. So we will get false results. Then there are certain mass analyzer parameters. Mass analyzer parameters for selected reaction monitoring. Selected reaction monitoring is a process by which we monitor the fragmentation of analyte to a particular fragment. The purpose is to ensure that to ensure that the analyte which we want to detect, that is only detected. There is a no mix and match. Now, so there are various parameters which are associated with the ion optics, which are used for the optimization of ion transport to the first quadrupole. And these parameters should be set as per the vendor's instructions. Vendor means whose mass spectrometer we are using. So we do a little variations in those parameters that suit to the individual compound. Uh, what precaution we have to take is that we have to avoid conditions that results in the fragmentation of the precursor ion, or that results in the excessive fragmentation of the precursor ion, means that ion which we are targeting. The other parameters that have an impact on quantification include is a mass resolution and calibration. So mass resolution was covered by Dr. Yolanda last day. And then there's another parameter, collision cell energy or gas flow and associated voltages. What is this collision cell energy? So first thing is, okay, what is selected reaction monitoring? Like if we want, this is particularly used when LCMS method is used for quantitative purpose. Now for quantitative purpose, we have to, for example, we want to determine the content of a ketoprofen. Now ketoprofen has a certain fragmentation pattern. Like ketoprofen has a molecular mass of 254. So in negative ESI mode, it will fragment to, it will ionize to 253, and it will fragment to 209 by the loss of CO2. So the primary fragment of ketoprofen is a 209 from 253. So this is a one reaction. This is a selected reaction from ketoprofen molecular ion to 209. So we have to monitor this. What we have to do, we have to just monitor the this conversion. We will see what is the concentration of we will see whether it is containing 253 and 209 as well, right? And this selected reaction monitoring is very much dependent upon collision cell energy. So see here the effect of collision cell energy. Collision cell energy, you understand that we are inducing fragmentation. So in quadrupole 1, it is analyzed. Then in quadrupole 2, it is collided. So what it will be, it will be collided with the electrons. So what is the energy of those electrons? So if those, see here, it is a 40 electron volts, it's a 50 electron volts, it's a 60 electron volt. This is a AGGC. This is a type of RNA, which is having a molecular mass of 1263, right? So as we are increasing the collision energy, see the number of fragments are increasing. So this is the molecular mass. This is a quasi-molecular ion at 1264, right? And here these are the fragments, and these are the fragments, and see here the abundance of your molecular ion. So molecular ion here, it is abundance is 800. Here the abundance of molecular ion is 300, and here the abundance of molecular ion is just 20. So molecular ion abundance will decrease, and the concentrate or the abundance of the fragments will increase. So what type of fragment, how much fragmentation we need, depending upon that, we have to set the collision energy. Then the other factor is solvent and pH of mobile phase. So in mass spectrometric analysis, the solvent, it has to be not viscous. Generally, we don't use chloroform because chloroform is heavier than water and it is a viscous solvent. Most common solvent used for LCMS is water, acetonitrile, and methanol. Because they are highly volatile, they are not heavier one. Preferred one is acetonitrile because methanol is viscous in comparison to acetonitrile. Methanol is viscous than water. And pH of the mobile phase, that is very important. pH of the mobile phase should be, see here, around 2 units, right, plus minus pKa. What is the pKa? 2 units around pKa. So if the pKa of the substance is, let's say, 5, then the pH of the mobile phase has to be from 6 to 7, so that it's ionized to the maximum extent. Then assessment of the empirical formula, we have to, once we come to know the mass by charge ratio, then from mass by charge ratio, we will detect the empirical formula. So this is specifically for qualitative analysis, where we want to detect the type of component which is present in it. It is already 1 o'clock. Can we continue it after the lunch? Because it will take a few minutes more. Or should we finish it?
So we can carry it after lunch.
Okay.
But contrasting, we can finish it.
Because after lunch again, I have to continue with the HPLC method development process. So, okay, we will continue in the after lunch, right? So see you at
Okay,
2.
sir. Fine.
So we have seen the effect of collision energy on fragmentation. So what information we need, whether we need molecular ion specifically, or we need the fragmentation pattern. So if we need the molecular ion information only, then we have to work at a lower collision energy. And if we want to need information about fragments or fragmentation pattern, then we have to work on the higher collision energy. Then the other factors are the solvent and pH of the mobile phase. So what kind of solvents are used? So as we have discussed that acetonitrile and methanol, they are the most commonly used solvents, organic solvents, along with water as aqueous. Between these two, acetonitrile and methanol, again acetonitrile is a preferred solvent because it is thinner in comparison to, or less viscous in comparison to, methanol. So so that the solvent viscosity does not hinder with the ionization process. Then on what is the polarity of the molecule? On that basis, we select a particular mode, whether it's a positive ESI mode or negative ESI mode. So if a molecule is acidic in nature, then we go for a negative ESI mode. If the molecule is a basic in nature, then we go for the positive ESI mode. Then pH of the mobile phase is important. So what type of what type of buffers are used for the preparation of mobile phase for LCMS specifically? Now, if we talk about the HPLC or UPLC, when it is coupled with the other detectors like UV detector, fluorescent detector, means all detectors other than this NMR and mass, then the buffer can be phosphate, buffer can be carbonate, buffer can be acetate, buffer can be formate. Means we can use any type of buffer for those purposes. But when we work on LCMS, then the buffer has to be volatile. Volatile buffers like acetate and formate and carbonate. Why? Because if we use non-volatile buffers, for example, phosphate is the most commonly used, or borate is the less commonly used. Then these buffers, they are not, the phosphate ions, they are not volatile. So in the mass spectrometer, they will not be destroyed. They will be sticking to the ionization chamber and to the mass analyzer as well, so resulting in contamination of mass detector or mass spectrometer as such, so hampering its sensitivity and selectivity. So what actually happens in a mass spectrometer, that the solvent gets evaporated. So when it gets evaporated, it means it is converted either to gases, right? It has to be converted to gases so that no solvent trace is left. Now, because phosphate is a non-volatile, so it will remain adherent to the ionization chamber, or it may pass on to the mass analyzer. So they have to be vapor, so have to be volatile. So we use either formic acid or acetic acid or ammonia or ammonium hydroxide or ammonium formate or ammonium acetate, because ammonium acetate ions, when they are heated to a let's say at 200 degrees Celsius or 300 degrees Celsius in a ionization chamber, they get degraded to like ammonium ion converted to ammonia, so that is a volatile, and the acetate ions, they get converted to this carbon dioxide and methane, so that they are all are lost. So these have to be volatile. Then pH. What should be the pH? Optimally, the pH has to be plus minus 1 of the pKa, so that the substance is ionized significantly, because ionization pH adversely affects the ionization. Then assessment of the empirical formula, that is another thing. Whenever we detect some component in a mass spectrometer, then from the molecular ion peak, from the M by Z ratio, what we have observed, we tend to find out its a molecular structure, and the first information which we can obtain from its M by Z mass ratio is a molecular formula. Now, how to derive that molecular formula? What are the different approaches? How we can narrow down onto the empirical formula? Right, the most helpful thing is the isotopic peaks. Now molecule, it has isotopic peaks like chlorine, bromine, and sulfur. Now see, the carbon, like it has a nominal mass of 12, but its accurate mass is a 12.0001. So this is the exact mass. So exact mass, and this is a nominal mass. So there's a one molecular weight, another category, another term is molecular mass. So what is the difference? Like chlorine. Chlorine it exists in two isotopes, chlorine 35 and chlorine 37. Now chlorine 35 is the mass of chlorine 35 isotope is 35, and the mass of chlorine 37 is 37, and the weight of chlorine is 35.5. Right, what is this? This is the average weight of chlorine, means it is you see averaged of all the its abundance of chlorine 37 and chlorine 35. So what is the weighted average of individual isotope? So based upon that, it's a molecular, its nominal mass is determined, so what we call it a molecular weight. So that is a difference. So in mass spectrometer, it is not the molecular weight, it is the molecular mass which is determined. So if in a substance contains a chlorine atom, so it can be chlorine 35 or it can be chlorine 37. So it will, if depending upon the abundance, it will detect both the masses. So accordingly, we will have additional peaks like M+2 peak, we call it. M+2 peak, like chlorine 37 is abundance is 35%, means 35% of the chlorine in this universe is 30, having a mass of 37, and the others are 35. So if a molecule is containing one chlorine atom, so there is a possibility that 33% of the molecules will have 37 mass of chlorine, and others will have 30. That's 35. So accordingly, there will be mass of mass detected for M with a molecular ion with 35, and the M+2 peak means where the molecule with the chlorine is 37, that will be also detected. So these isotope peaks, they help us in identification of the peak and identification of the molecular formula. Then we have to calculate the monoisotopic mass of the protonated and deprotonated molecules and the likely adductines. So what are these adductines? Like in ESI or EPS or chemical ionization, there is a possibility of formation of adductines. What are these adductines? Let's say we have dissolved, let's say mobile phases containing ammonium acetate. So, and we are working in a positive ESI mode. in a positive ESI mode. So M, the molecule will tend to accept one proton, right? But it can also accept ammonium ion. So there will be M plus H and M plus ammonium. So these M plus ammonium ions, they are called as adductines. Similarly, we can have M plus sodium, M plus potassium, M plus water, right? So depending upon what types of elements are present or ions are present in the mobile phase, those adductines are formed. So those adductines help us to locate the mass of a particular interest. This is the elemental composition calculator. This is a free software which is available, free app which is available on web. So if we let's say if we feed any accurate value up to 4 decimal or 5 decimal, right? And the usual tolerance value is fixed at 10. Tolerance means the deviation with respect to this. Like here, it is 0.0052. So the deviation is 0052 plus minus 10 means here 0.00542 0.0562. So this is the tolerance means the last last two digits. 10 means last 10 digits. So this is for software which gives you the possible molecular formulae, and out of these possible molecular formulae, we narrow down or we select the most appropriate molecular formula, which is fitting into our mass fragmentation or mass spectrum. This, we have taken an example where there are a few compounds. Trifluorine, malathion, right? So these are some pesticides which are detected. So this is their here precursor ions and product ions. Means precursor ion is a trifluorine, is the product ion is this. So this is the molecular ion and this is a product ion, right? Or we can say this is a precursor ion which is taken as a main ion from this compound, and this is the product ion which is monitored. So this is a SRM, selected reaction monitoring, right? And now for this particular product ion, there are some molecular formula which are assigned from this. So this we can see with the help of this exercise. Elemental composition calculator is there. I will show you how to use it. Let me share my screen, total screen with you. So is my screen visible? Yes, so it is visible to all. So see here, if we feed the exact mass of here, I'm feeding 305.1113, right? This one. And for this, because it is derived, if I calculate the molecular formulae, right? I am having these molecular formulae, numerous. See here, numerous molecular formulae. Out of these numerous molecular formulae, we have to select the most appropriate one. Now, there are the different strategies. Like, see here, trifluorine, if we see trifluorine, right, we know what is the molecular structure of trifluorine, and we know how many carbons are there, how we know how many hydrogens are there. And there is another rule, which call it a nitrogen rule. With the help of a nitrogen rule and with the help of index of hydrogen deficiency, which we call it the RDB. RDB stands for the ring, number of rings and double bonds. So it is index of hydrogen deficiency. right? So depending on the basis of that, index of hydrogen deficiency, index of nitrogen rule, we narrow down onto the a few molecular formulae, and with the help of fragmentation pattern, we can see this. So it is all possible molecular formulae, and if we see this C13H16F3N2O3, it is present here. Here it is. If you can see it, C13H16N2O3F3, it is a 305.1113, right? With a deviation of 0.002, right? So this is the way we can select the molecular formulae, and we can also take any other example. For example, you say here tetraconazole, for example. In tetraconazole, it is a 96.9901. So we feed here 96.9901, and we calculate. And again, we will see this molecular formula here, C2F3O. C2F3O. Here it is. So these molecular formulae are obtained from these softwares or these apps, but we have to apply certain principles which are very basic to the mass spectrometry. And with the help of that, we can identify the molecular formulae. Therefore, we can identify the molecular structures. This is another example. See here where they are detected phonophores at 500 microgram concentration of 500 microgram per kg in tomatoes. And so this is a full scan of this sample, where you are seeing several peaks, and when its chromatogram is extracted from this extracted ion chromatogram. So this is an extracted ion chromatogram. This we are showing, you see two signals. Two signals are shown here. One with the signal to noise ratio of 103, another signal to noise ratio of 88. And when this peak is fed into the mass, and we see what masses are present, then it is here where the mass analyzer plays a role. And it has shown you see 109 and 168 as its fragment peaks, and its molecular ion peak is here 198. And it's see its a theoretical mass is it's the practically mass observed is a 168.9920, but mass error is a 2.4 ppm, means the deviation is 2.4, means it is a accurate mass is 9916. Accurate mass is 9916, and its accurate mass is 109.0117, but detected is 0.0124. So there is an error of 6.4 ppm. So there will be error always. The error will be always there, but there is a limit to it. Maximum permissible error is 10 ppm. This is what we have done in elemental composition calculator. If I show you that I have, okay. So we had put a value of 10 in that application. Similarly, this is another example, tetrachlorovinphos, and here it is C again detected in tomato, and it has a mass error of 4.8 ppm, which is again within the acceptable limits. Then if we, so this is a mass, right? And if we go to the MS, and if we MS means 125, this 125 is set to the collision cell, second quadrupole pole, and you get a mass. So this is the precursor ion, and this is the product ion. It's a 78.9593, 68.962. This. So these are the product ions of this precursor ion. And here again, the mass error is just 3.8 ppm, which is within limits. Then in addition to these routinely followed applications, there are some recent advances like SRM and MRM in quantitative analysis. SRM is a selected reaction monitoring and MRM is a multiple reaction monitoring, which is used in quantitative analysis. And another is ion mobility. Ion mobility is latest development in mass spectrometry, which is used for speedy analysis of samples. So where number of samples are very large, so their ion mobility systems are used. So this is an example of selected reaction monitoring, like ketoprofen. We have taken the example. Its a molecular mass is a 253, and its product is a 209. So reaction conversion of 253 to 209. This is a reaction. So we have to monitor this reaction, and see here 209 is found, right? Another is 197. So these are the two fragments which are formed from ketoprofen. So what we are monitoring? We are monitoring 209 and 197. So if we are monitoring this, if we are seeing that these two are present along with small concentration or small abundance of 253, that sample definitely contains ketoprofen. So this ketoprofen is found in a wastewater effluent at a concentration of 50 nanogram per ml with this approach. So thank you so much for your attention. This is about mass spectrometry tutorial which we had discussed. So if you have any query or any doubt or any suggestion, please feel free to raise. There is a no query. Okay, so we start with our next topic of the day, which is about HPLC method development. So we will discuss here about the various issues which are related to HPLC method development, the precautions which we have to take, the conditions which we have to optimize, the ways we have to select a column, ways we have to select the mobile phase or solvent, the ways we have to select the operating temperature, way we have to select the tubings, or so on. So these discussions will help us to understand how we can develop a good HPLC method. So starting with that, so the total outline of this session is it was supposed to be in three parts. One is means there are three factors which affect the method development process. One is the selectivity, second is the efficiency, and third is the sample preparation. Means the first target is to increase the selectivity of the method. Means how much selective our method is, how effective it is to identify or to separate all components in a sample without any doubt. Then part two is efficiency, means how we can increase the sensitivity, how we can increase the resolution, right? So these will be discussed under efficiency. And the third one is the sample preparation. So this part I'm not going to touch because it is already covered by Dr. Ashok Malik, and it will be further covered by Dr. Holak. So chromatographic resolution, it's a major factor which major parameter which defines the success of any chromatographic method or HPLC method or UPLC method is the resolution and selectivity. So resolution is it depends upon the selectivity. So if we see this equation which we have seen yesterday also, so you must be remembering from that, resolution is dependent upon three factors. One is efficiency, second is selectivity, and third is retentivity. Efficiency means it is related to the number of theoretical plates. So directly with the particle size that we have already done. Then selectivity is dependent upon how the two peaks are separated from each other, and means what is the difference in their retention times, relative retention time. And the retentivity means how good they are retained. Then see here, if this is the initial one, if we increase the number of theoretical plates, then the efficiency is increased. Means if you increase the efficiency, the resolution is increased. If we increase the K, means the retentivity. So retentivity is increased means we have made the analytes to be retained for a longer period of time. So therefore, there is a delayed elution, and delayed elution results in the resolution, right? So resolution. Now, but the peaks are broad. Peaks are broad, and to increase the resolution, what we have to do, we have to make them sharp, and this can be increased by increasing the selectivity. So selectivity is increased, and therefore the peaks are also made sharper in comparison to the upper one, right? So by doubling the efficiency, we have achieved a 20 to 40% resolution. And with the double of retentivity, there is a further improvement of by 15 to 20%. And with a double of retentivity, selectivity, it is an improvement is by more than 400%. So if we recall the resolution, now why do we do chromatography? Why we go for the chromatographic separations, right? Maybe I think the entire reason we do chromatography is because of this equation. Our chromatographic resolution of the chromatographic experimentation is surrounding around this equation. At the end of the day, we are trying to separate or resolve components in a mixture. So that is the ultimate aim of our chromatographic experiment. And if we look at this resolution equation, we see the factors that affect the resolution are efficiency, selectivity, and retentivity, right? So this is we have seen in the last slide also. We can break these into two categories, right? So this is the efficiency. Efficiency is they are broken into two categories, right? So the one is the mechanical contribution, and second is the chemical or physical contribution. Now, what is a mechanical contribution? Mechanical contribution means which is related to the hardware, and chemical and physical contribution which is related to the software. So here there is no software, but there is a interactions. What kind of interactions are taking place between solute and solvent and stationary phase? So those are classified under physical, chemical, and chemical contributions. And finally, at the we see the impact of individual component on the resolutions, how the resolution is improved, right? So mechanical contribution, it is related to the particle morphology, means what is the particle shape, particle size, the packing of the columns, how well they are packed, in what way they are packed. Then physical and chemical contributions include the information about the complementary bonded phases, like if we have a normal phase silica or reverse phase silica, then multiple particle substrates, means there can be mixed phases. Then ability to utilize high pH, because usually we don't go for the high pH, but if we can modulate these, we can go for the high pH as high as 8. Then increase retentivity. So that is the ultimate aim of studying these factors. Now, chromatographic resolution, if we say have a closer examination of the general equation, general resolution equation, so that it reveals that there are two major categories, chemical and physical, right? So physical contributions are like the last example of the changing column length, and optimizing the chemical contribution is more challenging. This is see why it is a more challenging, because it is dependent upon this chemical is dependent upon analyte properties, it is dependent upon stationary properties, it is dependent upon mobile phase properties. So management of the interaction among these two properties. So this is more challenging. And whereas this efficiency, to increase the efficiency, what we have to do, we have to simply decrease the particle size, or we have to increase the column length. That's it. So chemical one is more challenging. So selectivity. So there are two factors. One is selectivity and retentivity. So let's see the selectivity, how we can improve the selectivity of a method in a reverse phase system. Now, this selectivity, if you see as a triangle, right, there are, you see the three, it's a three pronged issue. So one arm is a mobile phase pH, second is organic solvent, and third is the column chemistries. So selectivity can be enhanced by balance between, by choosing appropriate mobile phase pH, by choosing appropriate organic solvent, and by modulating with the or column chemistries. So we will see one by one the each factor affecting the selectivity. So coming to first the mobile phase pH. The pH, this is see, pH of the mobile phase is largely governed by the pK value of the compound which we are going to analyze. So at this here pH, what pH we have to select for at the beginning, right? And for that we follow the rule of plus minus 2. What is plus minus 2? So whatever the pK is plus minus 2, right, that should be the pH which we have to adopt for its method. If the pH is 4, then we have to adjust the pH within 2 to 6. We cannot go beyond 2, less than 2, we cannot go beyond 6, right? So if we adjust the pH plus minus 2 units from pKa, right, we will make 100% of the molecules either ionized or unionized, right? So that is the principle. If we are making the unionized, it means we are increasing the retention, and if they are ionized, we are decreasing the retention depending upon the stationary phase. Now let's see how the retention is dependent upon pH. So in this here, if see, there are three types of compounds: unionized, which are black in color, acidic, which are red in color, and basic, which are blue in color. And this is how the retention factor is affected by pH of the different kind of compounds. Like acidic compounds, they have higher retention at lower pH, and as the pH increases, the retention decreases. And conversely for base, as the pH increases, the retention increases. Means at this particular pH, like for acidic compound, at acidic pH, all the components will be unionized, and here all the solute will be ionized, and here all the basic components will be ionized, and here all the basic components will be unionized. So depending upon what is the pKa, we choose appropriate pH. And if we see the neutral one, they are not affected by pH. You take whatever the pH, so they are not affected. So let's take an example here. Let's say we have again 3 types of compounds: neutral, acid, and base, right? And if we record, if we run these 3 types of compounds at pH 2 with the mobile phases 2, then you see here the B component, which is a basic, right? It elutes almost with the mobile phase, so it is not retained at all, because at pH 2, B is converted to salt. It is highly soluble, so it is moves freely with the solvent. Whereas A becomes unionized, so it gets retained on the stationary phase and it is delayed. And the neutral has its own characteristics, so it is at around 1.7. And if we record the pH, if we run the same sample at pH 5, so see what has happened. Neutral is at the same retention time, right? And A and B both are at the 1 point, right? Because at pH 5 here, they are having the same characteristic, same ionization. So this is the isobestic point. Isobestic point is the pH at which the ionization is same, right? So this is the at isobestic point. So that's why they are eluting at the same retention time. So their ionization capability is same. If we further increase the pH, right? Say what happened, we made the pH alkaline here. So here the neutral is at the same pH, same retention time, right? And now here at alkaline pH, the base will be unionized and the acid will be ionized. So acid will elute first and base will elute later. So with the change in pH, what we have seen that the neutral species, they do not change their position, their elution pattern does not change, but if there are acidic and basic compounds, then it is greatly affected by pH, right? So pH is a very powerful selectivity tool. So whether we want to delay the elution or we want to make the component elute earlier, right? We can select a particular pH. Now this shows how the retention map can be used to predict compound movement, which can help to create selectivity. The curve for 4 different compounds are plotted here. 4 different compounds are B1, B2, A, and N. So B1, B2 are basic, and A is acidic, and N is neutral. The retention factor K for the compounds is shown on the y-axis, and on the x-axis is shown the pH scale here. At pH, see here we have a pH 2 and pH 10. So we have recorded at the 2 pH values, right? So at the pH 2, see here at pH 2, which is acidic in nature, so both basic components B1 and B2 will be ionized, so they will elute together, because both B1 and B2, if they are ionized, so their lipophilicity does not play significant role, so they will elute together as one peak, and acidic component will elute later. And if we made the pH 10 here, now here the acidic component will be ionized because acid-base reaction, and nitrogen remains the same, and B1, B2, they will be having because different retentivities, so they will be resolved because they are now unionized. So by suitable control of pH, we are able to retain a particular solute, and we are able to make a particular solute to elute ahead of the others. So pH is useful as therefore very powerful selectivity tool. So this is again the same. So small, this one is new 4 to 8. So what should be the range of our pH, range of pH of our mobile phase for optimum HPLC method development? It should be, let's say here it is 6, right? So it is an isobestic point, right? Isobestic point is 6. So 2 units lower than this, 2 units higher than this, and we get 4 to 8. So our this is a range, right, which can be exploited. So this is a very large range actually, 4 to 8, right? Usually if we change the pH unit by even 0.2 units, right, it shows enormous effects on the retentivity. So when we have selected this range, right, 4 to 8, and the isobestic point is, let's say, 6. So well, let's say we have recorded it for 5.5. So at 5.5, see B1 and B2, these two are slightly distinguished. They are not having the same retentivity because their ionization is slightly different now, and therefore they are resolved. And at pH 6, the acidic component and the B1 component, they have the same ionization capability. So they will elute as a one peak, whereas a B2 is having different, right? And N and B2, they are different. So here, see at pH 6, the retentive selectivity is less. At pH 5.5, selectivity is greater. So with the 0.5 units. So similarly, depending upon the pKa and pKb values of the substances, we can have enormous effects on this selectivity. So this is another example where we have seen why this, we can see that why pH control, why pH control is critical. Now changes in the mobile phase pH can have a dramatic effect on retention of this ionizable species. And so as per the general rule, we have seen that acidic compounds are retained most under acidic pH conditions, and the basic compounds are best retained using the high pH conditions. In both cases, the charged groups on the analyte is in its most neutral form, giving maximum retention. Ideally, the mobile phase pH should be 2 pK units higher or lower for retention for basic compounds, for acidic compound. For basic compounds, it should be higher than pKa, and for acidic compound, it should be 2 pH units lower. Here the neutral compounds, because they do not contain any ionizable group, they are not affected by mobile phase pH. So here, see, if we can see by changing the pH from 2 to 7 to 12, there is a huge change, huge mashup in the peaks. So one, right, pH 2 to 7, pH 1 is here, right, and 12, it is has moved further, right? So acidic components, so this is basically a basic, we can say acidic component. So this is a basic component. So at pH 2, the basic components are eluting earlier, and in acidic pH, in basic alkaline pH, the alkaline compound, basic acidic components, they are eluting earlier. So this way, by controlling the pH, we can make the components to resolve in a better way. And the neutral one, they are not changed. So this number 2 is a neutral one. Its position remains same. All other positions, it has changed significantly. So this is an example where we are seeing shift in the peaks because of shift in the pH or change in the pH, and that change in pH is you see very, very small. pH 5.0 to 5.2 to 5.4 to 5.6. So we have three components, paratoluene, lidocaine, and ibuprofen, and all are having a different chemical behaviors. Like toluene is a neutral, and lidocaine, it is basic in nature, and ibuprofen is acidic in nature. And at pH 5, they are well resolved, and at pH 5.2, 2 and 3 they merge. This one, they merge because their ionization becomes closer. And at pH 5.4, the peak number 3, it comes closer. So peak 2 and peak 3. So here peak 3, 3 number ibuprofen, with the increase in pH, the elution of retention of ibuprofen has reduced, and retention of lidocaine has increased. So by just 0.2 unit change in pH, the retention behavior of the two component has reversed, right? 2 coming earlier here, and here 3 coming earlier. And with further change in pH, increase in pH, right, the 3 will be eluting earlier and 2 will be retaining. And if we further increase, the 3 becomes, submerged with a neutral. And if we further increase the pH, right, it can come before 1, right? So this is how we can modulate pH to increase the selectivity. Now, so for good reproducibility, the +-2 rule is strictly followed in HPLC method development. Now, there are some practical pH range guidance for method development. So for new method developments, aqueous buffer should be in the range of 2 to 3 for acidic analytes, and pH 9 to 10 for basic analytes. And at these pH, they are when mixed with the organic solvents, most analytes will be unionized, and therefore better and more reproducibly retained for better selectivity. So if they are retained, their peak will be sharp. If they are sharp, then the resolution will be better. Now, while selecting these pH 2 to 3 or 9 to 10 or whatever, we have to first understand, we have to keep in mind the pH stability of the stationary phase, because it is possible that our stationary phase may not be stable at that particular pH. Usually, stationary phases are not stable at pH higher than 8. So if you are using pH 9 to 10, it may lead to column bleeding, right? So resulting in the column loss. So before starting selecting this pH, we have to know from the vendor's instructions for that particular column, what is the maximum and what is the minimum pH which can be operated in that column. So the second component for creating selectivity is column chemistries. Column chemistry, when we talk about column chemistry, we talk about the column, stationary phase characteristics, its pore size, its pore shape, and its a chemical nature, its a physical nature, right? So that we can select an appropriate stationary phase for method development. So packing material forms the stationary phase for chromatography. So these packing materials, they are filled into the stainless steel tubes to form columns, and they are packing material particles. They may consist of one of the following, like the three characteristics are there, like pores. What is the pore of that pore size of the particle? What is the pore shape of the particle? And then what is the surface area of that pore? Then what is the shape of the particle, whether it is a regular, regular or irregular? Then what is the bonded phase? Bonded phase means whether it is bonded to any other ligand or not. So accordingly, we have three types of particles. One is a porous, then fully porous, and then superficially porous. Superficially porous. These are different types of uh, particles. They have their own nature in terms of their chemistry, in terms of their physical nature. Like non-porous, they eliminate the mass transfer kinetics, and they have poor retention and mass loading. Because they are non-porous, it means they will not absorb anything. So whatever is happening on the surface, so it means they have a poor surface area or lesser surface area. Therefore, mass transfer kinetics will not be possible. What is mass transfer kinetics? So when the this is happening, this illusion is taking place when the solute is moving over a stationary phase under the influence of mobile phase. uh, it is moving from stationary phase into mobile phase, then from mobile phase into stationary phase, and then in stationary phase from outer layer to the inside of the pores, right? So this is a mass transfer, right? And the rate at which this transfer is taking place, we call it a mass transfer kinetics. Now, if it is a non-porous, it means whatever analyte is adhering to the particle or adhering to the stationary phase, it is just a surface phenomena. It is not getting into this. So mass transfer kinetics are almost absent, and because the surface area is less, therefore poor retention and poor mass loading. Then it can be fully porous. These are the most common liquid chromatographic particles. Like silica particles, they are usually fully porous, and they encompass a wide range of pore diameters and pore volumes. So depending upon their manufacturing practices, like you can see, there are numerous pores, right? So there are numerous pores, and so we have different particles which are having different pore diameters and different pore volumes, right? Then other third type, which one is a recent one, superficially porous. What is superficially porous? Means we have a one solid core, and around that core, this is a non-porous core, and around that non-porous core is adsorbed, is a a porous core. So this is a porous to this much extent, and but this is a porous throughout, and this is not porous at all. So these superficially porous particles, they have poor back pressure in comparison to fully porous. So for these particles, there are several parameters which we need to understand. One is the pore diameter. Now, pore diameter means, let's say this is our particle, right? And on that particle, there are several pores, and we have taken one pore which is lying here, and this is how does it look like. So this is a pore, and this is a pore volume, right? And this is how your silica looks like on that pore, right? So this is the silica which is responsible. These are the function groups which are responsible for the retention of solutes. So if we have an analyte whose molecular weight is more than 3,000, less than 3,000, then the recommended pore size is 60 to 130 angstrom. If the analyte weight is 3,000 to 10,000, then the pore size is increased 125 to 200. If the analyte weight is more than 10,000, then the pore size is 300 to 1,000. And if it is very large, then it has to be non-porous. Now, why with the increase in molecular weight, the pore size is increasing? This is because if, let's say, we have used this particular pore size for a large molecular weight, so large molecular weight, its size will be also greater, so that will not fit into this pore size, so it will not be retained. So for it to be retained, it must enter into that pore so that it can interact with these function groups through several interactions. So therefore, the pore diameter of the particle depends upon the molecular size. So accordingly, we have selected a column. So fully porous and solid core particles are available in a wide range of pore diameters. So see here, this is a 125 angstrom diameter, this is a 200, and this is a 430. Now add a 450 angstrom. So as you see diameters, the pore size is increasing. As the pore size is increasing, the shape is also changing. The outer surface, that is also changing. The importance of pore diameter, see here on a practical example. So here 1 and 2, 2 components, which is thyroglobulin, and this is the thyroglobulin dimer, and this is the thyroglobulin monomer, right? So this is they are not resolved. So it is a 200 angstrom. It is 450 angstrom, 450, right? So they are resolved, okay? And if we have mixed the two, right, this is a hybrid one, 200 and 450, then they are resolved to lesser extent, right? Why they are not resolved here? Because thyroglobulin dimer and thyroglobulin, they are high molecular weight substances. So see here, their molecular mass is a 1340 and 667. So they do not fit into 200 angstrom pore size. Therefore, they do not get retained, and they retain they elute very early, so they are not resolved. But with the 450s, with the 450 angstrom diameter, they get entrapped, they get settled into the pores, therefore they get retained. And similarly, if we see the myoglobulin and uracil, which are low molecular weight substances or intermediate molecular weight, so they, myoglobulin is 17 kilodalton and the uracil is 112 dalton. So they are again, see, resolved, right? So this is based upon their molecular weight and molecular size, and this retention. The second parameter about which affects the method development which guides the method development in HPLC is a pore volume and surface area. So this is the pore volume, right? And this is the total surface area. Now for that, we calculate the phase ratio. The phase ratio is the ratio of the pore volume to surface area. So what is phase ratio? It is calculated by this equation, area and the volume. And the base particle phase ratio means the ratio of the surface area to pore volume. It is primarily dictates the overall retentivity of the phase. Now differences, these are the different types of particles and their phase ratios. So we have a fully porous silica, and we have a fully porous hybrid, and here it is 159 meters square per centimeter cube, and here is a 126 meters square per centimeter cube. So if surface area to pore volume means the pore volume is, let's say, small, but the surface area is large, right? So the phase ratio will be large, right? And if it's a converse one, surface area is less and pore volume is greater, right? Then the phase ratio will be less. So this phase ratio, how it affects the resolution, see here, it is the two different stationary phases, one with a phase ratio of 159, another with a phase ratio of 126. So these 6 components, they are eluted with the same chromatographic system. And see here, when the phase ratio is less, the retention time, the total runtime is less, and when the phase ratio is greater, then the total retention time is total runtime is higher. So that indicates a greater efficiency of lower phase ratio. Then the fourth component is a particle composition. Particle composition means this surface, what is the surface characteristic of the stationary phase, right? So whether it is a pure silica or whether it is a hybrid or whether it is a bonded, right? So these are the particle compositions, different type of we have a silica particle, we have a polymeric particle, we have a hybrid particle. So they have the different characteristics or key attributes like pH range is for silica is 2 to 8, but polymeric is 1 to 14 and hybrid is 2 to 12. So with the polymeric is a synthetic one, right? So synthetic, they have a very wide pH range. So they are very, you see, rugged, and they are not affected by any pH, and they afford us to develop HPLC method using for same column can be used for a wide range of substances. Rigid silica particles are rigid, they are strong, and they are most widely used packing material. Whereas the polymeric one, they are pH stable in acid and base because the pH range is 1 to 14, and they are synthesized with the controlled pore size. And hybrid one, hybrid one is strong and rigid. They are pH stable in acids and bases, and they are again synthesized with controlled pore size, and they afford symmetrical peak shape. The disadvantage of silica is they are pH sensitive, so they dissolve at a pH greater than 8, and selenol activity results in a peak tailing in many basic analytes. These selenol groups here, this one, right? If they are not end capped, they cause peak tailing. And the polymeric one, they are not very rigid and they collapse under excessive pressure. And the hybrid one, they are none of the silica or polymeric material is having any disadvantage. Hybrid one. The application silica can be used for normal phase and reverse phase because it is a very rigid, very strong, and so it can exhibit, it can experience, it can withstand a very high back pressure. Polymeric one, which are soft, which are not very rigid, so they are used for size exclusion and gel permeation. And hybrid one, they are used for the reverse phase and high leak type of separations. Then particle size. Particle size is related to efficiency. So we have discussed a lot about it yesterday. About the particle size, how the particle size is affecting the resolution. By decreasing the particle size, we are increasing the surface area. By decreasing particle size, we are increasing a number of theoretical plates. We are decreasing the height equivalent to theoretical plates. So they all increase the efficiency and resolution, right? Then particle surface charge, right? So what is the what type of polarity it has? The surface has. See here the unbonded silica particle where the pore surface is having selenols. This is a normal silica where all silicons, they are having OH groups. So they are called as selenol groups. And these selenol groups, they are acidic in nature. So in acidic medium, they remain as such. But in basic medium, when the pH is 7, this selenol is converted, loses hydrogen, and they're converted to siloxide, SiO negative. So surface selenols, they are acidic site. At low pH mobile phase, so when the pH mobile phase is a low, that means it is acidic. So they remain uncharged and they behave as a weak acid. And if the pH of the mobile phase is alkaline, so they will be ionized, and selenols, they will get a deprotonate and they become negatively charged. So in acidic medium, they are neutral, they are a weak acid, and in case of a basic medium, they are negatively charged. So unionized or ionized. So that adversely affects the selectivity. So strong interactions between ionized surface selenols and basic analytes is promoted when pH is alkaline. This is C13 bonded and fully end capped high purity silica pores. So this is a bonded phase in which we have attached, we have modified the silica by attaching C18 chains on this. And because C18 chains are very long and the pore volume is less and pore size is also less, so it is not possible to fit to replace all hydrogens on the on the surface with this chains, because its size is very less, so it will not fit into this. So that leads to overcrowding. So this, because this overcrowding, some of the selenols, they are not removed, they are not covered with this, so they remain as such. And these selenols, they are residual selenols, they are responsible for tailing, because that will cause their surface properties will be affected by pH. So if it's a C18 and we are working with acidic pH, then it is okay, they will remain acidic, they will remain unionized. But if we are working at a pH 7, pH 6, this selenol will be deprotonate to selenoxide, and that results in a tailing, that results in either trailing or early elution, depending upon the properties of the particles of the analyte. Then we have mixed mode retention. Now, mixed mode retention is what is that? Now, this is a reverse phase interaction with the ligands. Here it is C1, 2, 3, 4, 5, 6, 7, 8. This is a decyl, means C8 chain. C8 chains are attached to this silicon. So as we have said, that all this OH will not be capped with, or will not be interacting with C8, so they will remain as such. And if we have this substance, tacrine, no, not tacrine, it's imipramine, right? So it is if it's a run in the mobile phase of pH less than 3, and this surface silicons, they will remain as selenols, OH, so they will remain intact, and there will be no charge on this, so it will elute without getting retained at this let's say point. Okay, now if we make the pH of the mobile phase 5, from 3 to 5 or more than 5, then OH will be converted to siloxide, right? And there are now, here you see siloxides are there, because these hydrogens are replaced, they are removed, right? And this is a negatively charged, and this nitrogen is having a positive charge, so there is an ionic interaction, so it will be retained. So if it is retained, it means its elution will be delayed, and see here, its elution is delayed from this to this. And moreover, there is a tailing, right? It is not a sharp peak. This is, you see, shark tail. This is because of this OH O negative ions, O negative oxides, they retain this through ionic interaction. So the elution becomes very, very slow. So this particle which is retained here, it elutes slowly over a prolonged period of time, therefore broadening the peak. We call it tailing. So basic compounds, they have high tailing when we use formic acid. Because formic acid is not a very weak acid, and so there is interactions between acid and base. So there is an unexpectedly high tailing factor for analytical mass load of basic analytes when we use low ionic strength acidic mobile phases. And this is due to reduction of the reduction of acidic selenols from high purity particles. Then the other type of particles are charged surface hybrid particles. Charged surface hybrid particles. This is let's see, a new technology in which some unbonded like silica particles are there. This is unbonded silica particles, normal silica particles. What we do, we apply some controlled surface charge. Means some cationic species are you see attached over the surface of this particle surface, right? So some add a reproducible low level charge to particle surface, and then it is covered with or coated with C18 or whatever we need to add. It can be C8, which can be phenyl, it can be pentafluorophenyl or so on. So it is a bonded and then end capped. And so it is a functionalized. So first it is a controlledly charged, and then it is a functionalized with the appropriate functional group. So the resulting particle is called as a charged surface hybrid particle. And this is the benefit. So they have two drugs, two compounds, quetiapine and propionone. See here, this is a normal, this is a porous silica, the conventional porous silica particle, and see the tailing observed in quetiapine. And then this is a core shell. Core shell is that in which there is a core, non-porous core, and over that we deposit a layer of silica to make it a core shell particle. And again here in that case, there is a tailing of the quetiapine. But when we make it CSF, means when it's a cationic and followed by a C18 chain, then quetiapine is very strong, very sharp peak, very sharp peak. So this is a possibility because quetiapine is a base, and it is CSF is a positively charged, so it is not retained. There is no ionic interaction between stationary phase and the analyte. So because there is no ionic interaction, so the retention is solely because of its polarity and not because of its ionizability, and therefore we get a sharp peak. So if during analysis method development process, we are continuously experiencing this problem that the peaks are not getting sharp, they are not, they are getting broad in all the possible columns or all the possible particles, then it's a time to shift to this charged surface hybrid particles. Then particle morphology. Particle morphology, its surface can be smooth, the surface can be rough, because as the particle size change, As the pore size change, the surface appears smooth to rough depending upon particle size. And how does it affect? There is no significant effect of particle morphology, because the only point is it has to be spherically, it has to be uniformly spherical. If they are irregularly shaped, then definitely it will affect the resolution. But if they are spherical, but their morphology can be, you see, surface characteristics can be shape, surface appearance is, let's say, rough or smooth, it doesn't make any difference. So last one is the ligand and ligand density. What type of ligand is attached to this silica? So depending upon that, we select a particular type of analyte. Like if it is a C18, so it is a use for general purpose, for balance, non-polar retention for acids, bases, and neutrals. So C18, it is applicable to almost all type of compounds. Then C18 has a mild coverage. Mild coverage means it is not fully, this is all sites maximally fully C18. This is intermediate one. All the silicons are not connected to C18. So it is for balanced retention for polar and non-polar compounds. Means both non-polar compounds can also be retained. Then we have embedded polar. So what is embedded polar? Between silicon and C18, we introduce some polar group like oxygen, like nitrogen, right? So that to increase the selectivity, right? To increase the peak shape, so on. Then we have phenyl hexyl, and it affords different selectivity, especially for aromatic compounds, because aromatic compounds, they interact with the aromatic system through pi-pi interactions, therefore making the aromatic compounds to retain. So if they're retained, then retentivity will be increased, and therefore the selectivity will be increased. Then we have a pentafluorophenyl propyl, and this is where all the 5 hydrogens of phenyl group, they are retained, they are replaced by fluorine. So we have different selectivity. They afford again different selectivity, especially for the basic compounds. And then we have a cyano column where the terminally we have one cyano group. So it is again it's a specific for polar compounds. This is a amide group here. See here, this is a amide group, and this is called as a hylic. Why hylic? Because it can interact with the polar as well as non-polar. So if it is a polar, it will interact with the NH2. If it is a non-polar, it will interact with the linker or carbonyl, right? So sometimes it happens that our sample contains only polar compounds. So if we are containing only polar compounds, then we have to select a mobile stationary phase which is polar in nature. And the most common polar column which is used in HPLC or UPLC is a normal silica column or amino column. Amino column means silicon's OH is replaced by NH2. Right, so that is called amino column. So they are amino columns or silica columns which are polar, because polar component, they will interact with the polar surface and they will get retained, and therefore they will get resolved. If our substance, our sample contains all non-polar components, then the stationary phase has to be non-polar. So that depends upon how much of non-polarity is there. If it is, we can use C18, we can use C8, we can use phenyl, we can use pentafluorophenyl, depending upon what type of compounds they are. And if it is containing a mixture of polar and non-polar compounds, then it is a tricky situation, because if we use polar stationary phase, and only polar will be retained, non-polar will not be retained, so they will not be resolved, and vice versa for the other. So for that particular site for type of compounds, the answer lies in the new type of columns, which are called amide columns or hylic columns. So they are having both types of characteristics. So they absorb or they retain polar components as well as non-polar components. So in summary, if we talk about the column chemistry, so the important column chemistry attributes increase the ligands and the particle substrate. So by having different ligands and different ligands which are attached to the particles, we can vary the column chemistry to suit every type of compounds. And it can be a reverse phase and a mixed mode behavior. Reverse mode would be like hylic is a mixed mode. Then second important information which we have obtained is consider the pH limitations of column. This is very, very important, because if we do not take care of this, we will be damaging our column. So that column will get destroyed. And the third component of creating selectivity is organic solvent. Now, there are different types of organic solvents which are used in chromatography, particularly HPLC and UPLC. And the most commonly used are methanol and acetonitrile, although the others are there which are used to little extent, like tetrahydrofuran, isopropyl alcohol, acetone, ethyl acetate, methylene chloride. So accordingly, there is a given elution strength. Methanol has a very weak elution strength, 1. Acetonitrile has a greater 3.1 than tetrahydrofuran, so on. And these are the specific informations like methanol and acetonitrile. They have very low toxicity and very low viscosity. Methanol is viscous than acetonitrile, but their toxicity, like methanol has a very low toxicity, acetonitrile has very low viscosity. But at the same time, acetonitrile is toxic. Depending upon what are our requirements, we can choose either methanol or acetonitrile. But at the same time, the handling has to be very careful, because every solvent, whether it is mentioned to be non-toxic, it has to be avoided the direct contact with skin. Then tetrahydrofuran, it is very rarely used, only under certain special circumstances, because it is usually contaminated with peroxides, right? Because tetrahydrofuran is readily oxidized by air, and it is also oxidized by air, so it is contaminated. So it is generally used only when there is a problem with the peak tailing. Isopropyl alcohol, it has a very high viscosity. Its viscosity is even greater than methanol, and it is used generally for the regeneration of stationary phases. What is this regeneration? Regeneration is that whenever we are using a column for a prolonged period of time, sometimes it happens that if the care is not taken properly, the inlet point of that column becomes permanently contaminated with the solutes, and therefore the back pressure increases significantly. And the back pressure increases to such an extent that we cannot use that column on a normal conditions. So for that, we have to remove those substances which are adsorbed at the inlet point of column. And for that, what we do, there is a set procedures in literature by which we run some solvents through the column, so that those in the reverse direction, so that the solutes which are adhered or strongly adsorbed to the at the inlet point, they get dissolved, they get dissolved in the solvent, and we can use that our column again. So IPA, acetone, ethyl acetone, methyl methylene chloride, they are generally used for this column regeneration. Acetone is also used in normal phase chromatography. Ethyl acetate is also used in normal phase chromatography, and methylene chloride is also used in normal phase chromatography. Now, when the mobile phases, the stationary phases, polar, and mobile phases, non-polar. So out of these, methanol and acetonitrile, they are the most commonly used. Methanol is a protic solvent, means it has a tendency to lose hydrogen, and it is more polar. Therefore, it exhibits a weak reverse phase elution. It is a weaker reverse phase elution solvent because it's a more polar, and it's a more viscous. Therefore, the lower flowing rate we have to adopt. We cannot work at very high flow rates when we are using methanol. Acetonitrile is a aprotic solvent which contains cyano group, so it's a more simple formula is CH3CN, and it is acting as only hydrogen bond acceptor, so polar interactions are taking place. And because this is containing a triple bond, so there's a pi-pi interactions also possible, and it is a less polar in comparison to methanol. So therefore, it is a strong reverse phase elution solvent. So this is how the selectivity, solvent selectivity affects the elution pattern. For example, Norge estimate and ethynyl estradiol. This is in methanol, and this is in acetonitrile. And because methanol is a weaker solvent, means it does not dissolve the components freely. Therefore, they are the solutes. They are eluted in a delayed way, right? So their runtime is last solvent solute has eluted at around 5.4 minutes, whereas acetonitrile is a strong solvent, so it elutes earlier, so 4.40. But there are certain, you see, or other issues like here, these two peaks, right? 3.5 and 3.7, they get mixed up, and this one, these two peaks, these three peaks, they also get mixed up in acetonitrile. So we have to select the appropriate solvent so that we can get maximum resolution or maximum selectivity. This is again the effect of solvent on 12 drugs, thymine, amiloride, doxylamine, or so on a column with the different solvents. Aqueous component is same, but the organic component is different. One methanol and acetonitrile, and see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. All the 12 peaks are there. Here are also all the 12 peaks, right? So this is with methanol, this is with acetonitrile. So all peaks are very well resolved, baseline resolution, but here number 1 and number 2 peaks, they are poorly resolved, and 11 and 12, although they are resolved, but they are very closely eluting, right? So this is so if the number of components in a solvent in a sample are very high, then we tend to use methanol, because methanol is not a strong solvent, so it allows the components to retain on the stationary phase, and therefore help in their retention, and therefore help in improving the resolution. So this is again another example with a different column. So in summary about the solvents, we see that acetonitrile is a popular reverse phase organic solvent because its strength is good, its viscosity is low. Methanol is a weaker solvent, which typically translates into longer retention times compared to separations with the same proportion of acetonitrile. But again, here the selection of methanol or acetonitrile depends upon the number of components and the polarity of the solutes. Methanol allows the pi-pi interaction between aromatic analytes and the stationary phase with aromatic groups on a phenyl column or a pentafluorophenyl, which can produce differences in selectivity. Then what are the, we have seen pH, we have seen organic phase, or right? So this the mobile phase is composed of these two things, pH and means aqueous component and its organic component. And organic component most commonly used as what methanol and or acetonitrile. Sometimes both the solvents can be used in a mixture. We make a ternary mixture, aqueous component, then also organic components, the two organic components. So mobile phase is important for best chromatography and the lowest baseline noise. So, but there are certain precautions or recommendations for optimum mobile phase. Use the highest purity organic solvents, water, and additives. So they are specially HPLC grade solvents or LCMS grade solvents which are available at the commercial level, and we have to use only those solvents. The reason is the normal analytical grade solvent, they contain impurities at the trace level, and those impurities, they affect the column life and therefore resolution. These solvents which are available commercially, they are not supposed to be filtered. Usually we are supposed to filter the water or buffer which we have prepared from 0.2 micron filter membrane. But these solvents are not supposed to be filtered because by filtering that we will be making them dirtier. So there is no need. So simply filter the aqueous phase and mix it with the required proportion with the suitable organic solvent to prepare the mobile phase. Then only dedicated bottles and other glasswares usually used for HPLC. Means let's say for HPLC there are or UPLC, they are reservoir bottles, and these reservoir bottles should be dedicated one. Like if we are using one bottle for methanol, it should be dedicated for methanol, one dedicated bottle for acetonitrile, one dedicated bottle for acetate or volatile buffers, one for the non-volatile buffers, one for the pure water. So they should be dedicated bottles so that there is no mix-up. Never wash HPLC glassware in a dishwasher with a detergent, so detergent is not used. Only rinse bottles in mobile phase quality water and solvent. The water which has to be used for HPLC or LC purpose, it is a special grade water. We call it HPLC grade water. So there are special water filters which are available in the commercial scales. Only those have to be used for all these experiments, even for the washings. Do not top off bottle with more mobile phase. Do not fill it up to the mark of the top. Old mobile phase will have changes in concentrations. Top of bottle means with once we have filled a mobile phase and we have run it for, let's say, 1 day, and at the end of the day, let's say we are left with 100 ml of mobile phase or 200 ml of mobile phase. Then if you want to refill that mobile phase, don't refill it in the same bottle because we have to discard the first mobile phase which was consumed, because as as the mobile phase is consumed, the headspace in the bottle increases, reservoir bottle increases, and that increase in reservoir bottle, increase in headspace causes evaporation of the organic component. Therefore, the composition of mobile phase change. So, if you, that's why we do not supposed to top off the bottle with more mobile phase. So we have to discard the old mobile phase, only then we have to fill the fresh mobile phase. Now, there is tools for optimizing selectivity in reverse phase method. So we have seen the three components: mobile phase, column chemistries, and organic solvents. These three components, they have their own role. Now, column chemistry is the one thing, and mobile phase pH and organic solvents, they are the others. So we have to mix them, and by optimum use of pH, by rational use of organic solvent, we can increase the selectivity. Then there are two other things which are used for optimizing the selectivity, that is temperature and gradient slope. Can we have a break for 5 minutes?
Sure, sir.
Thank you, Gaurav. So should we start? Okay. So temperature and gradient slope, these are the two other parameters which are very important in developing or in getting an optimized HPLC method. So how the temperature affects the method. Now temperature, we know that it affects everything. It affects every chemical process that occurs. So increasing the temperature will reduce the viscosity of the mobile phase. And this will result in lower system back pressure. So if the flow rate is held constant. So at a particular flow rate, with increase in temperature, the back pressure will be lowered because of decrease in the viscosity. And a high temperature will also change the ratio of partitioning between the stationary phase and mobile phase. Therefore, it improves the diffusivity of the analyte between the two, stationary phase and the mobile phase. However, due to this improved diffusivity, the optimal linear velocity is higher, and for the compound which are sensitive to temperature, which are sensitive to temperature change. Even a small change in temperature can provide a unique change in selectivity. So this temperature can affect in a multiple ways in the selection in the method development process. Now here, if you see the effect of temperature on separations, how separations are affected. So in the bottom here, we have a chromatogram, which is obtained at 30 degrees Celsius. And then we have a chromatogram at 40, 50, and 60 degrees Celsius. So as the temperature is increasing, the elution, the runtime is getting smaller, right? So from here, it is from 10 minutes, it has become 6 minutes at 60 degree. And the back pressure has also decreased from 1920 psi to 1160 psi under the used chromatographic conditions. And with increase in temperature, the number of theoretical plates have also increased. Therefore, the resolution is increased. So this number of plates have, how this number of plates have increased? Because we have not changed the particle, we have not changed the column, we have not altered the this particle size. Then how the plates have increased? Because of change in temperature, increase in temperature, the diffusivity has changed, and that diffusivity has culminated in you see quick mass transfer. That quick mass transfer is reflected in terms of theoretical plates. But these are the advantages of temperature in improving the separation. But there are certain disadvantages also. One disadvantage is that the column life becomes shorter, because at higher temperature there is a more accelerated chemical interactions. So the phases which are bonded to the silica phase, they will be affected, resulting in column leakage or column bleeding, and therefore column life becomes shorter. Secondly, in selecting the higher temperature, we have to keep in mind the thermal stability of the analyte which we are analyzing. So that is one limiting factor in increasing the temperature. Now, temperature and selectivity. See here, it is again the influence of change in temperature from 30 to 50 degrees Celsius. So there are now see 5 drugs: trimethylene, ethiazide, bumetanide, and benthiazide, and ethacrylic acid. So as the temperature is increasing, you see the elution is getting faster, and at 50 degrees Celsius, it has decreased the selectivity. So so what is the optimum temperature which gives us the selectivity? That we have to identify. It doesn't mean that as the temperature increases, we will get the selectivity. No. Selectivity, we have to optimize in a stepwise way, stepwise increase in the temperature. So like here, we are getting the good selectivity at 40 degrees Celsius, but not we are an unacceptable chromatogram at 50 degrees Celsius. So in short, the elevated temperatures, they are responsible for the earlier elution or the faster elution of most analytes. They reduce system back pressures, and they enhance the sensitivity also. Because how they enhance the sensitivity? Because if the see here, if the peak is eluting earlier, right, then peak is getting sharper. So as the peak is delayed, it becomes broad. As the peak is made elute earlier, it becomes sharp. So see here, this peak, and this peak. So this peak is sharpest, this peak is the broadest for among these three cases. So as the peak become sharper, width decreases, and as the width decrease, it becomes more and more sensitive. Sensitivity increases with the decrease in width. This we have seen yesterday. Now, change in the temperature, it also tend to change the selectivity of the separation, as we have seen in the last slide. And there is a possibility of, but at the same time, there's a possibility of separating the co-eluting peaks also, because solubility of components is greatly affected by mild change in temperature. So it is possible that under normal conditions, a normal temperature, two components may be having a similar solubility. But if we increase the solubility temperature by, let's say, 10 degrees Celsius, then the solubility of one component may increase significantly in comparison to the other, and therefore the two will get resolved because of change in their diffusivity. Okay, another component, another tool for optimizing the selectivity in method development process is a gradient slope. Now, what is the gradient slope? Now, in HPLC chromatography, there are two types of elution modes. One is isochromatic, another is a gradient. Isochromatic is that when mobile phase of one particular composition runs throughout the process, throughout the experiment. Let's say we have a total runtime of 30 minutes, and on that 30 minutes, let's say our mobile phase is a 50-50, water 50 and methanol 50 in a ratio of 50:50. So that mobile phase, moves, flows at the same flow rate, let's say 1 ml per minute throughout the 30 minutes. That is a isochromatic. Iso means same, right? But sometimes it happens that let's say that we have 7 peaks, and we are not able to resolve 2 or 3 peaks, 2 or 3 analytes with the help of isochromatic elution. Then what we have to do, we have to change the mobile phase change the composition of the mobile phase as it runs. Means, let's say when it starts, its composition is, let's say, methanol 50-50, and methanol 50 and water 50. And when it ends, let's say at that point, we want that the composition should be water 80% and methanol 10%. Now, methanol 20%. So in this case, we will select a program, we will tune the instrument so that the composition of the mobile phase will vary while it is running the run, right? So this is called gradient. Gradient means change, not the same throughout, it is a change. So there are different types of gradient. One is a shallow gradient, and another is a steep gradient. Shallow gradient means when the slope, shallow gradient slope means when the gradient change in the mobile phase composition is very slow. Means, let's say at 0 point, at 0 time, it is 50-50, and at, let's say, 30 minutes, it is let's say 60-40, right? So just a 10% difference. That is a very shallow change, right? Shallow gradient. And another can be steeper. Steeper is that, let's say, at 0 point, it is a 50-50, and at 30 minutes, it is, let's say, 100% methanol and 0% water. So this is a 50% change in the mobile phase composition. So this is a steep one. So the shallow gradient, it help improve resolution, but it decrease sensitivity, whereas the steep gradient, it helps to compress the peaks and oftenly reduce the resolution, but it increases the sensitivity. So it depends on the requirements. If we tend to improve the resolution, then we have to go for shallow gradients. And if we want to improve the sensitivity, then we have to go for a steeper gradient. Now, changing the gradient slope is a balance between peak height relative to resolution. So what is our requirement? Whether we want resolution only, or we want sensitivity only, or we want both, right? So we have to strike a balance and choose an appropriate slope, right? May not be too shallow, may not be too steep, so somewhere in between. And this gradient, it also causes changes in the retentivity and selectivity, because the mobile phase composition is changing, therefore retentivity and selectivity will change. Now this is a typical linear gradient, right? See here, at the x-axis, you are seeing 0 to 50. So let's say there is a solvent B, right? It has at a 0 time, its proportion is 0, and at, let's say, 50 minutes, its proportion is 50% over a period of 43 minutes. So on y-axis is the time shown. So this is a linear. So it is a linear proportion, linear increase. So this is a linear increase, right? And this is a segmented gradient. What is a segmented gradient? This segmented gradient is often done in the preparative chromatography. Preparative chromatography is a type of chromatography where we want to separate the components in large quantities. One is analytical, another is a preparative. Analytical is if we want to just analyze the component, what type of components are there, right? Then it is a analytical. If we want to separate the components in a larger quantities for some other use, then it becomes preparative. And for that, we have to use larger columns and we have to use larger pumps, right? And we need other specialized instruments for the recovery of the component from the element. Now, in segmented gradient, what is there? It is, you see, gradient is not uniform throughout. Let's say from 0 to 15 minutes, it is a one particular mobile phase composition going on. Then at 15 minutes, the composition will change abruptly to another composition, right? And for over a period of time, for example, 0 to, this is a column volume percentage, this is a time. Okay, here y-axis is time. So up to 20 minutes, the B phase is let's say 15%, and then from 20 to here 20 to 25 minutes, this B becomes from 15 to 35%, right? 20% increase. And then from 25 minutes onwards, it up to 50 minutes, it becomes 50%. So 35 to 50%. So this way, there is a different mobile phases at the 3 different steps. So different 1, 2, 3 types of columns. So why we carry out the segmented gradient? Because when we carry out we want to improve the resolution or we improve the sensitivity, we tend to improve the sensitivity or resolution in a particular segment of the chromatogram. Let's say here the resolution is okay, here the resolution is also okay. Only in this part there are, let's say, 2 or 3 peaks which are not acceptable, which are not having resolving. So we tend to increase use this gradient only in this particular part of the chromatogram, right? So it is called segmented gradient. So It is often done in the preparative chromatography because here we want to actually improve the resolution, and it inserts shallow fragments around compounds of interest. So it is a shallow one. It is a shallow one. It is a steep, then shallow, then steep, right? Purpose is to improve the resolution. See here, this is a linear methanol gradient at 60 degree, right? This is a linear gradient. So we have 1, 2, 3 major peaks, and we have small peaks here. And if we make it a focused gradient, right, then it becomes, then its solution is here. Now here, if you apply the focus gradient here, right, as we see in the lower graph, right, so we can achieve the better resolution, because here resolution is okay, all right, and here resolution is also okay, right, but resolution between first and second peak is not good, so we apply the gradient here only so that it can be resolved. So gradients, it's not necessarily that gradients have to be linear. So in the chromatographic system, whichever we are working, or whatever the company it is, whatever supplier it is from, they all have several gradient programs ranging from 1 to 11, and generally it is used 2 to 10 gradients, right? So 1 to 11 is not used. 2 to 11 type of gradient. So 6 number gradient is a, let's say, linear one, and 2 to 5 number of gradients, they are convex curves, and these are the concave curves, right? So this convex curves, they tend to shift forward, means here, means in the beginning it is steep, and then it is shallow. And these one concave one, these in the beginning they are shallow, and with the time they becomes steep. So what they serve different purposes. Different purposes how? The convex curves, they tend to shift the peaks forward. Because of change in this gradient, they will bring the peaks forward in the forward direction, right? And because at the later part of the chromatogram, it has become shallower, so it will stretch the later part, right? So in the beginning, the peaks will elute earlier, and in the later part, the peaks will elute late. And in the concave part, the earlier peaks will stretch, and the later peaks will compress, right? So this helps him to reduce the runtime also, the concave curves. So depending upon what type, in which region of the curve, which type of the chromatogram we want to apply gradient, or we want resolution, accordingly we can use convex curve, convex gradient, or concave gradient. So see here, example, this is a some components which are resolved, right? And if we apply curve 4 for this, then we apply curve 4 for this one, right? And the resultant elution is this way, right? So it has curve 4. Initially, it has made the peaks to elute earlier, means they will decrease the retention time. So see here, from 1 to 1.5, all the three are eluted, right? And here it is again at 1.5 to 2.0. So 4, it is yes, concave, convex, and this one. This one are significantly delayed. So these 5 are significantly delayed, right? Now here, these are also delayed. These peaks are also delayed here with respect to this, right? Now in the last slide, we have seen that the convex curves, they cause earlier elution of the earlier peaks, right? This one. So as per rule, these peaks should be eluted earlier than here. But it all depends upon, again, what is the composition of the mobile phase, right? So that also affects whether it will be shifted significantly or not, right? So ultimately, I see the later peaks have shifted, but with this illusion, the resolution between among these three peaks have increased, right? But the resolution among these later peaks has reduced. So again, we may need to apply some other gradient. So this is how it is fed into the program. Let's say at in the beginning of the time, right, up to 5 minutes, you see the mobile phase composition. A let's say stands for water, B, A for, let's say, for water, and B, let's say, stands for organic component. So 90:10. So over a period of 5 minutes, let's say A becomes 5 and B becomes 95. Then for 1 minute, it will remain same, and then for another 0.1 minute, it will come to 90 and 10. So this is how it does look. So 1 to 5, it will be increasing from 10 to 95, and then it will retain for the same, maintain, and then it will come back to the normal. So they are the different programs which we can use. So depending upon, see here, gradient must separate the critical peak pairs 4 and 5. So what is the purpose, right? Means whether we want to increase the resolution or sensitivity. Usually we apply the gradient to increase the resolution only. So gradients, in summary, by performing shallow gradients, we can improve resolution. And by segmenting the gradient, it is possible to maintain separations in critical illusion zones and in the selected illusion zones, so that we can reduce the time and solvent consumption in the runtime. And the changing gradient slope is a balance between peak height and a relative to resolution. So we have to maintain the balance. And the gradients, it's not necessary that they have to be linear. They can be non-linear. They can be concave and they can be convex. So this was the first part of method development, that is a selectivity. The second one is efficiency. How we can increase the efficiency. The efficiency can be increased by increasing the resolution. So this part we have already covered in the yesterday's session, where we had talked about UPLC, which is based upon the particle chemistry, and that improves the resolution. So efficiency is more as a physical phenomena, which is related upon column length, particle size, mobile phase flow rate, and instrumental performance. So this is again the same. The resolution is directly proportional to particle size, number of theoretical plates. So this is a comparison of fully porous and solid core columns. So here, solid core columns, this one, right? This is a solid core column. This is a solid core column, and these are the fully porous columns, right? And see the particle size, see here, it is decreasing from lower, from 5 to 3.5 to 2.5 to 1.7 to 1.6. So it is decreasing. As it is decreasing, the efficiency is increasing. So that is, we have seen in the last session. And again, we yesterday we had also discussed that with increase in flow rate, the resolution increases, but it is not a linear relationship. Up to a certain limit, the resolution increases. Beyond that, the resolution decreases, because that results in collapsing of the peaks. That results in a quick movement of solutes, therefore resulting in co-elution or close elution. And the solid core columns means where the solid core is having a porous particle adhered around it. So they have a better efficiency, because the reason is in the solid core, in the fully porous, the mobile phase will go up to the center of the pore, right? Center of the particle, right? Whereas in case of this solid core, it is only the surface phenomena. Let's say it is only 1.1 angstrom, right? 1.1 angstrom is the thickness, right? So it is mass transfer is only along 1.1 angstrom. But in case of solid fully porous, let's say the total diameter is 7 angstrom. So it will be around 7 angstrom. It will travel total 7 angstrom, right? So there is a mass transfer is more effective in case of solid core columns than in fully porous. So more effective is the mass transfer, more is the efficiency. So this already we have seen a Van Demeter curve. What is the relation between plate height and resolution and flow rate? And this is again, we have seen many examples in the last class. To select a proper efficiency, to achieve proper efficiency, we have to select a particular column or particular length or particular particle size, depending upon the number of components which are to be eluted, depending upon the nature of the components which are being eluted. Then efficiency can be also increased by turning chromatographic bands into peaks. So this is a broad band, which is a broad peak, we will say, not band, it's a broad peak. And this is a less sensitive, and this is a broad narrow peak, which is a more sensitive. So as we have said earlier that as the peak width decreases, sensitivity increases, right? So it is a less sensitive, this is a more sensitive because its width is less. Now, this is called dispersion. This phenomena is called dispersion. The analyte has dispersed along a certain width. Why this dispersion has taken place? Now, there are two reasons. One is the dispersion which can take place in the column, and second is the dispersion which takes place outside the column. And in the column, dispersion takes place because of eddies diffusion, because of the linear diffusion, right? Because of the mass transfer, right? Which are related to this Van Demeter equation. And in the outside the column, it can be due to this tubings, right? The tubing which is connecting the sample to column, then column to detector, then what is the detector cell, what is the shape of that, what is the flow dwell volume of that, right? What is the dwell volume of a pump, right? So these all affect the efficiency. So there are extra column factors or column factors. So the column factors, they include end fitting design, packing bed uniformity, column volume, particle size, right? And in extra volume, they include the other instrumental contributors like injector, like tubing, like connectors, detector cell, or data capture rate. Like this is a tubing. Now, this tubing, which is a connecting column to the detector or injector to the column, their diameter is very important. If the diameter is small, then they give you sharp peaks, and if the diameter is large, they will give you broad peaks. Because if the diameter is small, there will be no diffusion. And if the diameter is large, there will be large diffusion, therefore the peak will become broad. So larger is the diameter, broader will be the peak. Then connectors. Sometimes we need to connect. When we connect the tubing, connect the tubing with a column, right, through a connector, right? So it is sometimes it is possible that a gap exists. Ideally, this tube and this tube, they should completely align, right? Whatever comes from here, it should enter into this, right? It should be completely aligned. There should be no space around it. But this happens because when we cut the tubings of appropriate length through by using using non-conventional or non-standard methods, right? So if we have to use standard methods, so there are commercially available tube cutters. So if we use only those tube cutters, this problem will not be there. But if we don't use tube cutters, we use some other local methods, then that result in this kind of situation where the two ends of the peak does not match, and that result in you see here eddies diffusion and causing band broadening. So we have three types of systems: HPLC, UHPLC, and now there is another UPLC, which is ultra pressure liquid chromatography. So dispersion is see here. Here the dispersion is more than 30 microliter. Here the dispersion is 12 to 30, and here the dispersion is less than 12. So with UPLC, what we are doing, we are using narrow broads. The flow rate is fast, and tubing is also small, right? So therefore, their dispersion is less, their sensitivities are also greater. So this is a comparison between HPLC, UHPLC, and UPLC. See here, column length is same, right? Diameter is different, right? And see the efficiency here, right? And resolution. So the resolution has significantly increased. So all three are good. So it depends what we available have. So in short, this extra column or instrumental contributors and column contributors to band spreading, they must be minimized so that we can get narrow peaks so that the sensitivity is enhanced. And the instrumental contributions, they become easily important as column dimensions decrease, because as the column dimensions decrease, the eddies diffusion or the other contributing factor, they become almost zero. So whatever the contributions are there, they are from the instruments. Thank you for your patient hearing. If you have any query or any doubt, we can take on.
Hello, good evening, sir.
Good evening, Ji. Good evening, Gaurav.
Sir, my many congratulations for presenting such a well-designed and highly informative lecture on method development with HPLC.
Thank you, dear.
It was highly organized, comprising every minute details of HPLC procedure, and it was truly remarkable and appreciable, sir.
Thank
It
you.
will be highly beneficial for experienced as well as beginner on HPLC systems. It was perfect, sir. So again, many best wishes, sir, and many congratulations for presenting such a beautiful lecture.
Thank you, Gaurav. But I would like to have some discussion on it. If there is some query or some, you see, critical inputs from your side, that will be more appreciated.
I think, sir, you have covered almost all the aspects. Nothing is left. So I don't think so there is anything to ask.
Okay, so let's see if there is any other participant who want to, they have some another query.
Yeah, sure, sir.
Yeah. So there seems no response from any other participant. So we take the leave today, right? So we wind up our session today here only. And tomorrow, the session will start at again 10 o'clock. So I request all the participants to join by 9:55 sharp so that the expert is not asked to wait, right? So it doesn't look nice because we have invited them, and if they are to wait, it doesn't look nice. So we request you to at least join and in time so that we don't waste time on that, right? Thank you so much. Have a great day.